location. I also have elemental composition data. So this is a graphic representing the C to N ratio of sediment organic material, root, and shoot tissues. So the red line that you see is the proposed eutrophication threshold, and it's a bit counterintuitive when you're looking at it. Anything under the red line is considered eutrophic, and anything above is not. So the blade tissues, or the shoots, tended to be more eutrophic than the root tissues, and the sediment organic material was highly variable. I also have stable isotope data for nitrogen-15, and the goal with this was to try to do source tracking, but that is actually very convoluted, and that is because a lot of the ranges of values provided for the nitrogen-15 isotope overlap. So what I mean by that is, for example, septic effluent will usually have values for the nitrogen-15 isotope of between 3 and 5. Whereas fertilizer is a bit more depleted with a range of values between 0 and 3. There's a little overlap there, but not too much. Once you add in autochthonously produced nitrogen, like nitrogen fixation, the values are more depleted and range from -2 to +2, which completely overlaps with fertilizer and makes it a little bit complicated. Nevertheless, the sediment data that I have here appears to show that it has a nitrogen source associated with septic effluent, and the seagrass tissue data that I have, while predominantly influenced by nitrogen fixation goes on within the rhizome of seagrasses, which is right at the sediment and water interface. So certainly these seagrasses are impacted by the potential septic effluent source of nitrogen that exists in the sediment. There are a variety of implications of this research, one of which is that this study exposes the importance of pore water nutrient conditions for seagrasses and further defines what is good versus bad sediment quality as it relates to seagrass resilience. And this study also helps highlight areas of the southern IRL that are not suitable for seagrass restoration efforts, despite how attractive the sites might look at first glance. I'm sure all of us are very aware science never happens in a vacuum. I had a lot of help, and I'm really grateful to the people who gave their time and energy and money. That was very, very helpful. Are there any questions?
We have time for one quick question. Mike.
Yes. So the question was, with less seagrass, the nutrients levels could be higher because there's less uptake of those nutrients. That was the question, right? I mean, that's certainly there's certainly a possibility that that is the case. However, I do definitely believe that ammonium toxicity is a big factor in the fact that we have a loss of seagrass over time, especially because there have been previous studies where ammonium concentrations were about 20 micromolar and they were seeing effects of ammonium toxicity. And some of the sites that the data that I have is 70 micromolar or above. Yeah. I don't know if
Great.
that answers.
Thank you, Sarah.
Yeah.
All right, next up, we're going to leave the sediments, and Stacy Cecil is going to tell us about water quality to infer seagrass recovery in the northern IRL.
Hello, guys. So I'm Stacy Cecil. I'm with the St. Johns River Water Management District, and I tend to spend most of my time looking at water quality, our long-term surface water quality data set, but I also have the unique privilege to work with all the wonderful scientists who study seagrasses at the district. So we all know that the lagoon has had an unprecedented loss of seagrasses over the past decade, while in 2011 and 2021, we kind of saw our largest declines, as you'll see in this graphic on the side. There was a little bit of recovery in 2013 and 2015 for about the whole lagoon, but after the 2021 decline, really only the Mosquito Lagoon had significant recovery in the past couple of years. So I wanted to look at the lagoon and determine if there were any difference between SAV recovery areas and other regions of the lagoon by using our water quality data set, our precipitation data set, and some of the information we have on the change in land cover. So you're not unfamiliar with these graphs. You've seen them year after year, but they are a little different this time. They're organized by different regions of the lagoon instead of just taking the average across the entire lagoon. So I wanted to look at specifically the area between North IRL and Mosquito Lagoon because they tend to have some water quality characteristics that are similar, especially on the eastern side, and compare it to our areas that don't really seem to have a lot of recovery, the central Indian River Lagoon and the southern Banana Lagoon. So that's why they're lumped in a little bit a different way. Our seagrass extent are our bars on these charts, while our seagrass transect is the black lines, and our seagrass percent cover are the gray lines along the bottom. And as you can see from the two charts, we do tend to see a lot more of the increase in our extent, cover, and transect length in the northern regions of the lagoon than in the southern regions of the lagoon. In our precipitation, we're looking at annual averages from 1996 to 2024. And you can see that there are kind of cycles of wetter and drier years with the animation on the side. So the warm colors are our drier times, while our cool colors are our wetter times. And there tends to be a little bit of a dynamic between the north area of the lagoon being slightly drier than the central and southern banana regions of the lagoon. And that kind of poses a problem long-term when we're thinking about stormwater runoff and other implications of nutrient sources in the lagoon, because long, dry periods followed by instantaneous wet periods tend to cause conditions where nutrients can't be taken up rapidly by our vegetation or the normal sediment, natural sediment fluxes in the watershed. The other data set we're looking at is rate of urbanization. So you can see that we have three main comparable data sets at Access, one in 2008, one in 2014, and one in 2020. So as you can see from the 3 time steps, there is a roughly linear correlation in how we've become more urbanized in the lagoon over time, on top of the already rampant urbanization we've seen in the past. But you can also see from the graph on the side that we have a lot more wetland and natural characteristics in our northern areas of Volusia County, Brevard County border compared to the southern Banana River Lagoon and Central Lagoon being a little bit more urban. So you see the more wetland area in green, the more urban area in gray. And finally, to the water quality. These are only select water quality parameters that we kind of tend to focus on because of the urbanization question and the eutrophication questions we have. These also tend to be typical drivers of lower light availability in the lagoon and triggers for chlorophyll and the presence of algal blooms. So when we're looking at these, we're mostly going to be looking at biogeochemical and physical parameters. You can see from all 4 of these graphics, they kind of follow similar patterns across the 2 regions. So you can't really disseminate a great deal of information from just looking at a regular line graph to figure out what's going on in the lagoon, other than there's kind of a similar pattern. So that means to compare these large data sets, we wanted to kind of look at a permanova, or a permutational analysis of variance, with pairwise comparisons. Along the x-axis, you can see all the different factors we're going to be using in our analysis, from precipitation to land cover change to our nutrient data and some of our physical water quality parameters. The overall test that we ran did find a significant difference between the 2 regions that we're looking at in terms of seagrass cover and the rest of these factors, indicated by our very low p-value and our very high pseudo-F value. So there is definitely a difference between the 2 regions. And we brought in the pairwise comparisons to try to determine what might be driving those differences within the areas that we're looking at. And you can see that the t values on our y-axis do show the degree or magnitude of difference. But when we were looking at the p values of each of these, unfortunately, TSS and land cover change, at least in terms of seagrass percent cover, didn't really seem to make a significant difference, whereas all the precipitation and the other water quality parameters do. That's not to say that these aren't important factors, because when we look deeper into the pairwise analysis across all of our factors, land cover change does make an important difference for some of our nutrient information. So we don't want to discount it fully, even though it doesn't have a significant relationship with seagrass cover. But in the end, what does this all mean? Basically, our water quality is a very important driver along with our hydrology, so the precipitation, into the two different areas. So that's one of the things that we need to pay attention to in the future as we move along with our recovery strategies. But in order to tease apart some of those differences, we really need to run some additional tests and some additional models to help evaluate the weight of certain factors that are being used. Because the Mosquito Lagoon never really lost full seagrass cover, it's also entirely probable that the existing propagules in the seed bank that Isabel mentioned before play a really important role in the recovery of those areas. So in the future, kind of looking at additional modeling to be done and considering additional source parameters for some of our nutrients to help tease apart some of these differences as we consider different time steps. So as I mentioned before, this particular analysis only looked at annual averages, but I think if we expanded into seasonality, looking at wet season versus dry season, we'd get a little bit more information. I know that was fast, and I will leave you with a lovely video of our seagrass recovery in the Mosquito Lagoon that Lauren Paul has provided, and answer questions.
Questions for Stacy? Bob?
I'd like to speculate and have you comment on that. The Mosquito Lagoon is different in another aspect, it's bathymetry. Most shoreline slope in it, the seagrass recovery goes deeper. In the Mosquito Lagoon, it seems like it's a flat platform and virtually all the seagrass is at the same shallow depth. So if conditions become good, it explodes. Can you use your water quality at that depth for
Bob, your question was basically, can we use the bathymetry data to help insinuate some of the recovery from Mosquito Lagoon because it's a shallower depth than the other parts of the lagoon that we're looking at? This particular study didn't look at depth per se. We mostly have surface water data, but it would be very interesting to use the elevation in this way and add it to the model so that we can get a better picture of what's going on. Does that help answer your question?
Great, thank you, Stacy.
You're welcome.
And then last up for our 10-minute talks, we have Jordan Beckler talking about high-frequency benthic nutrient flux monitoring in the IRL. And we just want to give a shout out to Jordan for filling in at the last minute and probably doing this presentation over the weekend. So thank you.
I appreciate it. Yeah, so actually it's a bit of a bait and switch, I must say, because today I'm going to show data from a stormwater pond, but there is high relevance to the IRL, so forgive me. So I'm Jordan Beckler. I'm an associate research professor here at Harbor Branch. I'm going to present a story by my lead author, or the lead author is Mason, my student, Mason Thaxton. We just submitted this to LNO a month or two ago. And we're going to talk about a new system that we developed. I want to thank, obviously, our co-authors, but then also some additional students, some folks from FAU Engineering and Tech Transfer Office, of course, Harbor Branch Foundation for supporting this work, along with the Sensing Institute at iSense, EPA, and FDEP to some extent. Okay. So we know that sediments are important from the last few talks. Obviously, they can be responsible for certain conditions in the Indian River Lagoon. You can use benthic fluxes as indicators of sediment health, right? And benthic fluxes, sediment fluxes, they also tend to directly affect water column health. And there's a general lack of time-resolved benthic flux data. This review paper here from just a few years ago, they showed that interannual variability can be substantial. They say less than order of magnitude, but I mean, heck, even that's a lot, right? Time series benthic flux data is rare. You do get large and rapid responses to both internal and external drivers. So Fox and Treffry have done a ton of work looking in IRL muck, or organic fine-grained sediments, as they like to call them. Extremely sulfidic systems, intense sulfate reduction there. You can somewhat predict, or I guess you can, temperature is responsible for governing these fluxes. You, of course, do get lags and deviations. I've done a lot of work myself in the Lake Okeechobee continuum. There it's a lot of iron oxides, and you get all these crazy redox processes that are really governing inorganic nutrient fluxes, both ammonium and phosphate. So it's not really something that you can predict, with for example, temperature. So you really do need to make these time-resolved measurements. Something like harmful algae blooms. A bloom dies, incorporates all these organic nutrients, it ends up in the sediments, it's remineralized, releases those nutrients to the pore waters, they can then flux out. That can happen over weekly or perhaps even daily timescales. Finally, after restoration, dredging being an obvious one, right, it's pretty important to observe how the sediments respond. And so a common technique that folks use is benthic chamber incubations. It's basically a cylinder with a sealed lid. You put it on top of the sediments. At time zero, the concentration of whatever you're trying to look at in this overlying water parcel should be equivalent to the ambient water. And then as material accumulates from the flux or from the sediment flux into the chamber, you see a concentration change. You take this concentration change with time, account for your chamber geometry, and you could get units of flux. So that's sort of the basis of the measurement. But it's a ton of work to do this, right? It's a boat trip to deploy it, potentially a boat trip to retrieve it. Let's say 3 people. It's a lot of money, right? Realistically, you're not going to be doing this more frequently than typically it's like monthly, but I mean, even in theory, you could probably only get at best, let's say like weekly, consistently, realistically. The other issue is you're not discriminating between what's occurring in this water column parcel from what's truly coming from sediments versus the actual water localized processes, right? Photosynthesis and respiration still happening. This is important inshore where these waters can be dynamic. So we developed an in situ benthic lander platform called the Carousel. We deploy this to the seafloor, for periods of weeks to months. It operates autonomously. You see here a benthic chamber, and the benthic chamber rotates around to various what we call portholes, which are just small openings with a penetrator that enters the sediments. So it forms a seal on top of here, and you can incubate these sediments. So it's just like the previous slide, what I showed. It's just doing it autonomously and at different positions in the sediments. So we call that the total flux, right? You're getting water and sediment inputs into the chamber. We also seal off one of these portholes. So when the chamber visits it, you're just incubating water. So that's a water-only flux. If you subtract the two and do a little more math, it's a little more complicated than that, but you subtract the two, you can end up with your true sediment flux. So it's really just processes happening directly attributed to the sediments. Now we have a flow-through pumping system that's recirculating water. It's gently mixing the chamber, which is an important thing that you need to have a homogeneous chamber. In this case, we're just using a sonde. We're measuring dissolved oxygen, ammonium, and nitrate using ion-selective electrodes. These only work in fresh water. So we're getting these measurements over time. We deployed it in the stormwater pond this past July, directly across the street from where we are. We're using the Erlon site right here to get ancillary meteorological data. It is a low organic site, relatively sandy. There's really not much going on there, to be honest. Now, what we get over a 2-week-long period from the sonde measurements is just a continuous time series, right? The sensor is sampling the entire time. It's just the location from where the chamber is actually sampling from is changing, right? Sometimes you're doing the different types of incubations. So you do see these daily patterns in DO and temperature that you would expect, day-night cycling. For something like, nitrate, right, it's pretty variable. Some weird stuff going on there. Ammonium, we get this drift, and this is a product of using these ion-selective electrodes, but we can account for this by doing a pre-calibration, post-deployment calibration. It's really important to do matrix-matched standard addition calibrations, but eventually we can adjust this to get decent concentration data. When you zoom in, though, on this kind of noisy drift line, what you see is the noise is not noise. It's actually a product of the incubations. So when you're doing a total sediment exposed incubation, you see your concentration of ammonium increases. Water incubation, it increases not as much, right? Because you don't have that sediment input. Now the black is when the chamber opens up, you basically reset it. The chamber contents and what you're sampling become equilibrated with the ambient water. So you're still getting ambient time series water data, just not like the resolution you would expect if you had the sonde directly in the water. So when we calculate our fluxes from those incubation accumulation rates for dissolved oxygen, ammonium, nitrate, and we're looking at the water-only fluxes and sediment-only fluxes here, you see the water, because the chamber is clear. I forgot to mention that. The chamber is transparent, so we're allowing both photosynthesis and respiration to occur in the chamber. But you see this oscillation where oxygen is produced during the day in the water, it's consumed at night in the water. In the sediments, you're always consuming oxygen, right? The red line here. Ammonium, same thing. The sediments are always consuming ammonium. We do get, some oscillation in the water column, but what's interesting here is we had a rain event right after this period, and you can see, the nitrate, the ammonium fluxes, they do respond. I don't have time to go into that in detail, but we are seeing a response to environmental conditions in the benthic fluxes. When you look at time of day, this is an average of those those fluxes at different times of day. For the water-only incubations, obviously at night you have consumption, production during the day due to photosynthesis. Sediment, again, right? Only consumption due to respiration. There you see your ammonium, these hashed bars. It's being produced by the sediments. Nitrate, again, it's a little wonky. What we do know just by looking at red field and these fluxes of oxygen, ammonium, and nitrate is we have excess oxygen consumption, suggesting we have really sort of cryptic nitrification, denitrification going on. Oops. So I got to wrap up. It's the first ever automated time series ammonium benthic fluxes. I think we can claim that. We are discriminating between water and true sediment fluxes, and this is important because the flux is isolated to the water can be a very substantial component of the total flux that you would receive or that you would infer from a typical benthic chamber incubation. We use this multi-porthole design. It avoids artifacts from, say, repeatedly incubating the same sediment area over and over again. It also could facilitate in situ experimentation, right? You could add something like phoslock to 3 of those portholes and leave the other 3 open and kind of see how that might sequester the phosphate flux. It's a sensor agnostic design, so you could imagine outfitting it with sensors to monitor carbon fluxes. In this case, we're working now with a spectrophotometric nutrient analyzer as well, so we're shifting to a seawater environment. We'll get nitrate, phosphate, and ammonium here pretty soon in seawater. Of course, we get a wealth of data for statistics. If you're interested, I could share the paper with you. But, we're seeing that wind direction, wind speed, precipitation are all affecting these fluxes. Ultimately, what we're really after is being able to take sonde data. We deploy this system for a finite amount of time. We understand the environmental controls. And then all you got to do is deploy a sonde in the water like you regularly would, and you can then predict benthic flux data. So am I out of time? Okay, I'll stop there. A couple more projects right here in our backyard, and then in Lake Okeechobee coming up. So sorry.
Wonderful. Thank you, Jordan.
Time goes fast.
If there are any questions, you can catch them at the lunch break. So that's the culmination of our 10-minute talks. Let's just do one more big round of applause for everyone. That was amazing. Wonderful talks. And so now I'm going to pass the baton to Kristen, who's going to do their 3-minute talks.
All right, everyone. Before we move on to our 3-minute talks, I just wanted to give you all an update on the live stream. We did have a few issues this morning, and we've currently switched the link to YouTube. So if you know someone or someone's contacted you about the live stream, please have them email Indian River Lagoon Symposium at gmail.com, and we will forward the new link to them. So moving into our 3-minute talks, they are the only things that are standing between us and what's going to be an amazing lunch, I hope. Let's see. For those of you who have not done this before, just a reminder, I'm going to have a very short introduction for each of the presentations, and then the 3 minutes will begin. We'll have a timer here that will audibly go off after 3 minutes, so you all will know when 3 minutes is up as well. I suggest if you are next on deck, you come to the bottom of the stairs here and be ready to come up. I'm going to stay on stage here. And we're not going to have any questions now, but we can discuss them at lunchtime. And then Brian. We found the net. So if you go a little long, it will be here and available, and we're not afraid to use it. So if you just want to leave it there, yeah. Thank you. All right. So first up, for our first 3-minute session is, you want to come on up here? Because I'm going to start it. All right. Your timer. Oh, you got your own timer. All right.
Well, just,
Okay. So our first talk is going to be by Austin Fox. He's going to be discussing spatial and temporal trends for chronic short-duration hypoxia in the Indian River Lagoon.
Well, thank you to our organizers for setting this up so perfectly. I'm going to touch on things that relate to our previous 3 talks. And so over the past few years, we've been looking at hypoxia in the Indian River Lagoon. A lot of you are familiar with hypoxic zones like the Gulf of Mexico, the Dead Zone. In the Indian River Lagoon, in shallow coastal environments, we have hypoxia. It just behaves a little bit differently. It's this diurnal trend. So during the day, we have lots of oxygen. At night, it goes hypoxic. Jordan just touched on that a little bit. And so we've set out to try to investigate this, and it takes a lot of sites, right? So we have over 90 sensors out there at any given time, and looking at temporal trends, right? We can make 90 lines on a graph. It's chaos to look at, but look below the 2 milligrams per liter. At any given time, there's probably somewhere in the IRL experiencing hypoxia. And so we can now, with this many sites, we can start to look at spatial trends. We can make these beautiful maps looking at the spatial distribution of hypoxia. This is looking at 1 hour of one day. I can do this every hour for every day for the last year. Tons of data to deal with. Mary's going to talk about that coming up, how we manage all of that. So how do we combine these spatial and temporal things? We can now make these graphs looking at the percent of time that any area spends hypoxic, averaged over different amounts of time. So far, we're looking at it by month. But now we can start to identify areas that are experiencing frequent and extended hypoxia events, or experience, for example, 20, 30, 40% of the time are spent hypoxic. Thinking back about benthic fluxes, sediments, when they're oxic, they act as a sink for nutrients, right? So our keynote speaker today talked about assimilative capacity. That's what we're talking about. These sediments can take up and remove nutrients. When the sediments go hypoxic, they flip. They turn into a source of dissolved nutrients, right? And so where are we headed, right? Lagoon management, BMAPs are based on this assimilative capacity. And so we are closing in. We're honing in on being able to model the assimilative capacity of IRL. And so we're really close to a nutrient budget for the internal fluxes in the IRL, BRL. And so this touches on so many things, right? Hypoxia relates to and interacts with the infauna. The infauna bioturbate push that oxygen down to help keep those sediments oxic. And so just something that I always talk about is we talk about the fish kills, you hear about the fish kills, you think about those as our hypoxic events, but looking at the bottom, there are far more hypoxic events than you would know, than you could think about based on the fish. And so keep an eye out for those dead crabs. If you see them, send me an email. We're trying to keep a monitoring network of where are we seeing benthic mortality events. And so thank you to all of our sponsors. Thank you for so many people who have done this. And a special thanks to Dennis Anasek, who was such a great mentor for so many years. So thank you.
Right. Well, you have set a high bar because we are before 3 minutes. All right, so next up is going to be, Brian Botson, and he's going to be discussing leveraging Erlon data and PyCO2 sys to track down post-storm ocean acidification.
Thank you, Kristen. Extreme weather events can have profound effects on carbonate chemistry, as seen in these previous studies, which collectively demonstrate how storm events can disrupt carbonate chemistry by altering salinity, pH, CO2, and aragonite saturation state, thereby impacting the health and resilience of these ecosystems. To look at these kind of impacts in the Indian River Lagoon, we use Pyceotusis with input parameters from the Erlon network shown here to estimate aragonite saturation state before and after two storm events, Hurricane Ian in 2022 and Hurricane Milton in 2024, at 3 of our Erlon stations shown here. And then we used a machine learning approach, specifically random forest models, to determine what explained most variation in aragonite saturation state. These time series plots clearly demonstrate storm-related trends, including declines in salinity, pH, and aragonite saturation state, along with rising rainfall, discharge, and CO2 and turbidity. So this shows a significant decline in storm-driven in aragonite saturation state following storms, indicating the sensitivity of carbonate chemistry to storm disturbances. These patterns highlight the dynamic response of the system to storm events. Our analysis using random forest models identified key environmental factors, discharge, turbidity, rainfall, and wind gusts that influence aragonite saturation state during or following storms. Overall, we had a good model fit. And the most important drivers vary by storm, with turbidity being the most important during Milton, and discharge during turbidity during Milton and discharge during Ian. So we noticed a rebound in aragonite saturation state following storms, along with salinity and pH. And this highlights the resilience of the system, but also underscores the stress of continued storm events from climate change. Thank you.
All right, thank you very much, Brian. All right, so next up, we have from the University of Central Florida, we have Carla Persky with Seagrass on the Brink, Implications of Marine Heat Waves in the Indian River Lagoon.
Our oceans, lakes, and estuaries are warming at an unprecedented rate. This warming, in turn, increases the likelihood of marine heat waves occurring, which is prolonged periods of high sea surface temperatures. Globally, marine heat waves are responsible for up to 45% decline in seagrass cover, but the responses of seagrass to these warming temperatures may vary. For example, in the Mediterranean, Posidonia oceanica exhibited a mass flowering event. In contrast, these species from Australia, they completely died off. Here in the Indian River Lagoon, warming temperatures may trigger algae blooms, cause hypoxia, all which are conditions unfavorable for seagrass growth and reproduction. While many studies have been conducted across the globe, no one has examined these trends in the Indian River Lagoon, which leads me to my research questions. My goal was to examine the frequency, intensity, and duration of marine heat waves in the Indian River Lagoon. And we did this by looking at the climatology over the last decade at 6 different sites using the package HeatWave R, which utilizes a 31-day sliding window approach for the analyses. So what did we find? While some areas like Mosquito Lagoon at the north of this map exhibited mostly moderate events highlighted in yellow, all other sites stood out as hotspots for Category 2 strong events shown in the orange pie slices. I wanted to zoom in into the biggest pie size, which was SR528, or the Cocoa Beach area. This marine heat wave peaked in August of 2023. This happens to be the same year that we found flowers and seeds for the first time in the northern Indian River Lagoon. So there were some limitations to this study. This was only 10 years of data. For a robust marine heat wave analyses, we need at least 30 years of data. And so what I'm currently working on is these satellite-acquired data sets at the 5-kilometer pixel that will really allow us to have a better spatial extent of these marine heat waves. The implications of marine heat waves is that they may already be driving the flowering frequency and seed viability of these plants. So if we can identify these resilient versus vulnerable areas to these marine heat waves, we may not only slow their decline, but they may already be primed for survival in a hotter future. Thank you.
All right, up next, we have Mary McDonald. We have establishing methods to track short-term diel hypoxia in shallow, well-mixed estuaries.
Hello. So dissolved oxygen reflects many biogeochemical processes, making it a key indicator of water quality. An issue and obstacle that we can run into with hypoxia are these diel cycles influencing the day and night require both temporal and spatial resolution data to fully capture what these cycles are doing. So to address this, at Florida Tech, we've established processes and systems in an effort to better track diel hypoxia. What we've done is we've created a sensor network that is cost-effective. It is high-resolution data spanning the entire BRL, as you see here, and it is continuous temporal data with sensors from over 90 sites monitoring dissolved oxygen concentrations hourly. So how we manage and monitor these sensors is through these 4 steps. First, we have our sensor swap. We take lab-calibrated and launched sensors into the field, and we swap it with the retrieve sensor. Here's an image of a deployed sensor and one that we're retrieving from the field in housings. Our second step, we take these retrieve sensors from the field back into our lab, and we can read them out with mobile devices or with laptops, making it easy to access this data, calibrate it, and in this lab setting. For our third step, we do QA/QC procedures with quality assurance, and we're checking the calibration and verifying this data from the field in case any biofouling affected it. And for our fourth step, we take all this abundance of data from these 90 sites, and we compile it into a large managed database where we're easily able to access these 90 sensors, and we can make amazing informative graphs with it, as you saw before. And yeah, this data can be a lot. But with this, we're able to create high-quality contour maps where we can showcase both the high-resolution data and the temporal data, as you can see to the right, with times of 8 a.m. and 5 p.m., and being able to have this large spatial resolution with this, with just bringing more information into tracking diel hypoxia. With this, we can partner with restoration efforts across the IRL and also provide insight into management resources to better manage hypoxia. Further expansion into the IRL will provide high-resolution data, widespread continuous data, where we can better use this to manage hypoxia in a changing climate. Thank you for your time.
All right, up next, we have climate-driven shifts in fish communities along Florida's East Coast from Meredith Pratt.
Historically, water temperatures restricted marine fish ranges. However, climate change and shifts in water temperature are enabling some species to expand their ranges poleward. While this phenomenon has been explored in some regions for some species, it remains unclear what the implications of these novel species assemblages mean for coastal fish community populations. Therefore, we are investigating how Florida's East Coast fish community has changed over time and what potential environmental mechanisms may be driving these community-wide shifts. To do this, we used Florida Fish and Wildlife's Fisheries Independent Monitoring Program data, also called FIM. We used FIM data from Northeast Florida around Jacksonville, the Northern IRL, and the Southern IRL. Using the FIM data, non-metric multidimensional scaling of the annual fish abundance from 2001 to 2021 was used to visualize fish community shifts over time. We then ran generalized linear models to assess environmental drivers that may influence the fish community's species abundance and richness. The NNBS plots here show the annual fish community abundance over time for each of the 3 regions mentioned prior. The dark purple polygons represent the first 10 years in this study, where the light blue polygons represent the last 10 years. Each of the plots in each region show a similar trend, that fish communities have shifted over time regardless of location on the east coast of Florida, as indicated by the red arrows. What is different between locations is when the community began to become dissimilar. Jacksonville has a consistent shift over time, the Northern IRL has a break at 2010, and the Southern IRL has a very evident break between 2013 and 2014. The generalized linear models for each region were then used to determine what may be driving these shifts. Each region had a different best fit model, indicating that a one-size-fits-all answer is not the case. However, all of the models, except the Southern IRL's species abundance model, had year as a significant factor. The effective year was a positive one, resulting in abundance and richness to increase over time, suggesting long-term trends in species dynamics. Additionally, all of the species abundance models, other than the Southern IRL again, had temperature as a significant factor, explaining that higher temperatures had a negative effect on abundance. These model results indicate that longer environmental changes may have a larger impact on the fish community, as most of the models have included the temporal factor as well as varying environmental factors. Now, why is this important? As the fishing capital of the world, Florida relies on its healthy fish communities to sustain both its ecosystems and its economy. This research provides valuable insights on how climate change affects estuarine fisheries, which in turn can help inform policymakers and managers, ensuring effective conservation measures for species that are both ecologically and economically significant. Thank you all for your time.
All right, next up, we have Nicholas Curto, and he's going to be talking about oyster spat monitoring in the Southern Indian River Lagoon and St. Lucie Estuary, 2020 to 2024.
All righty, thank you very much for the introduction. And yes, I'm the first one to talk about oysters and spat today. So, I went too far ahead. Jumping right into it. So, I conduct monthly monitoring throughout the Southern Indian River Lagoon in St. Lucie Estuary. So I go to a total of 7 sites that you can see on the map to the right there. 4 of them are in the St. Lucie Estuary, represented by the squares, and 3 of them are in the IRL, represented by the circles. And at each of these sites, there are what we call oyster tees, eloquently shaped like T's, and there are 3 of them at each one. And from the oyster tees themselves, we suspend oyster stringers, which are in figure 1A right here, which have the shells suspended in the water column. And so every month we go out and collect the stringers from those sites and bring them back, and we count the number of spat that are on the shell there. So yes, a very scary graph. So I'm going to walk you guys through what's going on here, starting at the upper part of the graph here with the highlighted sections. So the highlighted sections are color-coded, as you can see here, with the red representing low salinity levels, which are very unideal for the oyster spat. The yellow represents higher salinity levels, which are still not favorable. The green is what I would call the Goldilocks zone, the 10 to 28 parts per thousand. That's like the oysters are loving that. And then the zone above that in the orange represents salinity levels that are a little higher than what you would typically find spat wanting to recruit in, but still all right. And the lines that you see going through each of the sections represent each year from 2020 to 2024. So as you can see, looking on the one at the right with the St. Lucie, it tends to kind of fall within that Goldilocks zone a little more, while with the IRL, it falls within that more higher range. Shifting focus to the lines at the bottom here, the colors represent each year, the same as it does with the salinity. However, each of the points represent the spat, that's the 28-month average. So basically, That's the number of spat that is found at those locations that I previously mentioned at both the St. Lucie and the IRL. So the main thing I want you to take away from looking at this data is spat numbers are really low at both locations. So compared to locations such as the Chesapeake Bay area, which has seen a profound resurgence of spat, it's up to 80 at some of those locations in the Chesapeake, while at most of the locations in both the IRL and the St. Lucie, with the exception of that one point there back in 2022, it almost never gets above 20 most of the time, with 2024 being the worst year out of all the years so far. So essentially, I'm advocating to be the canary in the coal mine, and if you have any questions, please find me afterwards.
All right, to close out our first 3-minute, 3-slide session, we're going to end with Taryn Chera and the presentation is Leveraging Mosquito Impoundments to Treat Eutrophic Waters in the Indian River Lagoon.
Thank you. Hi, everyone. My name is Taryn Chea, and I'm a PhD student from UF Whitney Lab. And my dissertation research is focused on using existing mosquito impoundment infrastructure to be leveraged to improve the IRL's water quality, similar to a constructed treatment wetland. So treatment wetlands use natural wetland processes like denitrification, plant uptake, and low-flow settling basins to remove suspended solids, nutrients, and other pollutants that are traveling from an influent point to an outlet point. And so comparing that to mosquito impoundments, there's some similarities there. Mosquito impoundments are essentially diked wetlands that have a pumping station that's pumping water into the wetland, and that would be the inflow point. And then there's culverts located around the perimeter of the impoundment that act as outlet points, and those are modulated or used to modulate water level for mosquito control. So what if there's a certain management regime that improves nutrient removal? And that's kind of what I'm trying to do through my research. And so in 2023, I started to monitor an impoundment to understand how management and site location affect water quality and nutrients. So I've been collecting water quality and nutrient measurements on a near-weekly basis at these 3 sites that are on this map. We have the pumping station, which is the orange circle. That's where water is being pumped in. And then the 2 squares are culverts, where the water can exit. And the most notable nutrient shift that we see is in inorganic nitrogen. So these graphs are showing some of the average monthly data that I have so far, with ammonium on the top and nitrite on the bottom, both in milligrams per liter. And results so far suggest that nutrients are increasing during the cooler months. And this coincides with culverts being opened, as well as a lower water level. And this is in accordance with rotational impoundment management, which is used by most of the impoundments that are managed in this region. So with this management scheme, water levels are intentionally raised in the summertime to limit mosquito hatchings, and that's done using the pumping station and the culverts. And then in the cooler months, the culverts are opened, and this can typically cause a water drawdown to occur. And so this seasonally dependent management regime that's currently being used is showing some sort of influence on the system's biogeochemistry, and we're continuing to monitor this specific system to see how nutrients are transforming in this system. And so I also have ongoing monitoring at other impoundments in collaboration with Brevard County Mosquito Control, and that will help us understand how impoundment management and characteristics are affecting soil and water as we're looking at them spatially and temporally. So thanks for listening, and I'll be here today and tomorrow for questions or comments. Thank you.
All right, and with that ends our morning session. So just a few things before we start our lunch break. If you need to know what we're going to be eating, we have menus at the beginning of each of the lines. You can also scan one of the QR codes to see what the menu is. Also remember, if you get a chance, to go out and visit the RV Sunburst outside. They're doing tours of that. And finally, don't forget to vote for our People's Choice Awards. So that's ongoing. Again, that closes at 7 o'clock tonight, but please vote for those as you can. And am I missing any? Yep. If you want to let the students try and go first so they can go to their posters, that would be really great too. Testing, testing. Welcome back, everyone. This is your 2-minute warning. Welcome back, everyone. We're going to go ahead and get started. Please go ahead and take a seat. Thank you. Before we get started with our next section, Rachel is going to make a short announcement.
All right, everyone. So thank you for your patience during lunch. I know it was a little slow. Sometimes, you know, in science and in life, when you try things differently, they don't always go exactly how you hope. But if you didn't get enough food, we will have pizzas being delivered during the break in about 45 minutes. So if you're really hungry, we'll make an announcement when the pizza comes, and feel free to grab a piece and come back in. We don't want anyone starving today. So thank you.
Thanks, Rachel. All right, let's go ahead and get started and get you closer to that slice of pizza. So I am very happy to be kicking off our first after lunch session, simply titled Fauna. Of course, that's near and dear to my heart. And starting us off today is a talk from Alexis. She's going to talk about zooplankton behavioral studies in the IRL.
in situ stereo imaging system for zooplankton behavioral studies in the Indian River Lagoon. So for this project, I'm focusing on zooplankton and micronectin, which range from tens of microns to a few centimeters in size. And these are really key components of aquatic food webs, linking phytoplankton to higher trophic levels, and include groups such as copepods, krill, larval fish, jellies, and small shrimp. And currently there exists this research gap where there's extensive studies on phytoplankton and higher trophic levels, such as fish or marine mammals, but really there's this missing link of zooplankton and micronectin represented. So really this is because they're difficult to quantify using standard imaging techniques because they're too small, or traditional sampling such as IOP measurements where we could see chlorophyll A. So really there's this need for a high-resolution, non-invasive imaging to study these spatial patterns and behavioral in a natural setting. So to propose a solution to this, we want to use stereo vision. So stereo vision essentially uses binocular vision like our eyes, where we can use two cameras to position an object in 3D space. So this starts with 5 main components, and it starts with image acquisition, which is taking a synchronized image from 2 cameras, and then going into image rectification, which is making sure the images match on line by line, then matching the features in each image up, calculating the depth from the 2 images, and then plotting this on a 3D map. So going into the stereo vision system that we developed, this stereo vision system includes 2 synchronized high-resolution 33 frames per second cameras with interchangeable lenses that allow for adjustable field of vision. And we also are using an external red-white illumination source so we can switch between the 2. And we have custom housings where each camera is separated into its own enclosure, and this allows us to adjust the camera distances as well as their angles. And overall, this is a very lightweight system with a housing depth rating of up to 100 meters. So going into our first process step is calibration. So for calibration, we did test here on campus in the TTC engineering facility, and we used a checkerboard pattern in order to map the differences and discrepancy between the 2 cameras that we're using. And we used the MATLAB stereo calibration application to be able to match these 2 and be able to position each cameras in a 3-dimensional volume as seen on the right here. So these validation tests included using 16 dried shrimp on a board in order to determine and see these in a rectified image, which is where we're matching them up line by line. And then going through with a larger window interrogation model where we're going to find our objects of interest, in this case the shrimp, and then going through with a smaller window interrogation to find our final disparity map where we can see our shrimp a lot more easily identifiable in the right image there. And then we're using our disparity calculations along with our calibration metrics that we did previously to generate a depth map so we can actually translate that and validate it with the actual depth positioning that we used in that test. And then you can see the zoomed in depth map with our region of interest there. And this allows us to resolve particles in our closest in-focus planes from hundreds of microns to 9 centimeters, and then from our farthest in-focus plane to 450 microns to 15 centimeters, which is exactly what we're looking for to study these types of species. So in the Indian River Lagoon, we took a video of a ctenophore. And what's interesting about this system is we're able to extract individual characteristics, such as 3D positioning and the speed and acceleration of an individual organism, which we can see represented in the graph above. The ctenophore in this video is remaining relatively stationary, but another way we can represent this data is to plot it in a 3-dimensional space. So here is an example of that plot where we have the 3D tracked position of this ctenophore over 260 frames, right? And with the color, we see the time progressing, where it's starting off remaining relatively stationary and then transitioning a little bit, but overall in a relatively stationary positioning. Then we move on to another video that we collected of a fish school in the Indian River Lagoon. As you can see, we can see some interesting characteristics here. The 2 videos are the left is our primary and secondary cameras, which is really just our left and right cameras. In the first process, we do some pre-processing techniques to be able to extract the features of interest out of these images so we can bump up our contrast, so it's a lot easier to identify the fish here. And then we can identify the characteristics we care about. So for this one, we care about nearest neighbor distances, or NND, which we can see in the left plot. So this is also in a three-dimensional space, so the lines you see here represent the distance in 3D. So we're only seeing a 2D representation right here. And on the right plot, we see their orientation. So we see how the fish are positioned respective to each other. This can be really important to know, like, how are they orienting themselves in respects to flow, or how they're organizing themselves in group dynamics, and can be applied to a variety of studies. Looking at this on a graph, the important thing to take away from this is we saw similar results to other studies, but this is really just a small time scale where we saw the average nearest neighbor distance was decreasing when we saw fish density or the individual amount of fish increasing. So really, as they were getting closer together as there became more of them. And really, another thing that we looked at was polarity and polarization order. And these are just like historically used parameters that we can use to determine the organization of this fish school. So we have a polarity of 0 or polarization order of 1, and that's a really organized school. Whereas if we have a polarity of 90 degrees or polarization order of 0, that's a random school. And really, this nearest neighbor distance and orientation data can provide insights into how our aquatic organisms navigate their environment, respond to various stimuli, and interact with each other. So in conclusion, we were able to successfully capture 2 videos of a ctenophore and fish school in the Indian River Lagoon, demonstrating its potential in other zooplankton and micronectin studies. And we were able to generate a 3D depth map of approximately 1,500 centimeters cubed underwater, and this offers a clear spatial representation. For zooplankton tracking, we were able to effectively track across a size range from a few millimeters to tens of centimeters. And in the future, we plan on focusing the image processing pipeline and to enhance all of these qualities towards smaller species like mysid shrimp, as well as improve the tracking across a variety of environmental conditions, such as the turbid Indian River Lagoon environment. Thank you all. Slide messed up a little bit, but I'd like to thank everybody involved in this project, as well as my various funding sources and the staff at the Harbor Branch Oceanographic Institute Machine Shop. Thank you.
Great, thank you, Alexis. That was awesome time management. So we have a chance for a couple questions. Any takers? All right, well, maybe you can meet up over a slice of pizza with Alexis at the break. Thank you. All right, next we are going to hear from Andrew, who's going to talk to us about sawfish in the southern IRL.
All right, thank you. So this talk is focused on the movements of big sawfish in the IRL. And when it comes to sawfish, big is pretty relative. So to introduce you to some of the size classes that we talk about, we have YOIs, young of the year, and small juveniles, anywhere from about 2.5 to 6 or 7 feet long. And we would consider those to be small. And then large juveniles between about 7 and 11 feet, and adults from 11 to about 16 feet would be considered big for the purposes of this talk. So we're not going to talk about the little guys, but you can stay tuned in a couple talks, and my friend Sarah will be talking about the small juveniles in the IRL as well. But we're going to focus on the big ones. So since 2016, we've caught 42 individual big sawfish in the IRL. One of those we actually recapped just a couple days later. And what you can see on this graph here is that our early sampling effort from 2016 to 2021 was all opportunistic. So there's only a few captures there where our collaborators managed to get their hands on a few fish. But in 2022, we started to do our own sampling and came over here and started to set our drum lines and use rod and reel to target these animals. Most of the fish that we've been catching have been those adult size fish from about 11 or 12 to upwards of 15, 15 and a half feet. And the vast majority of the sawfish that we're catching in our sampling are females as opposed to males. And when we look at this by month for all of the sampling that we've done, we see that females are definitely more common, and males are generally pretty rare. But we don't really catch a lot of females in the spring, We also don't do a lot of sampling in the spring, so we want to kind of continue this effort and see what happens. But we see this obvious peak here in August, and that is mostly attributed to the fact that local charter captains and fishermen had been contacting us through our encounter hotline, which my boss Greg will talk about in a little bit as well. And they were just telling us repeatedly over years and years that, hey, the second week of August, you guys might want to come over to the IRL and try to catch these fish. And they were definitely right. When we look at the length frequency of the animals that we're capturing, we see the females on the top. Again, those are mostly adult females. Very rarely do we catch any large juvenile females. And the males, although the sample size is much smaller, there's quite a bit larger distribution of sizes that we catch with them. But the focus of this talk is really, what are they doing once we tag them? So this is kind of a general overview of some of the fish that we've caught. And we have time on the x-axis, and they're ranked kind of in order of their stretch total length on the y-axis, with the largest fish at the top and the smallest fish at the bottom. And the general idea here is that sawfish that are tagged on the east coast of Florida stay on the east coast of Florida. They spend a lot of time over here. Those are denoted by the blue circles. But if you look really closely, you can kind of see that these sawfish are heading south to the Keys in the wintertime. So for the 2 years really that are denoted here, you see them going down south and spending their winters in the Keys. And then there's a couple of circumstances here where these fish are actually being detected on the west coast of Florida as well. So there's only a handful of fish here where we see that switch from the east coast to the west coast. But that's interesting because when we look at the fish that we normally tag in our normal sampling on the west coast of Florida, we've put out over 300 tags in sawfish on the west coast, and only a handful of them have actually been detected on the east coast of Florida. But you see the same trend, just the colors are basically inverted on this plot. So the sawfish from the west coast tend to stay on the west coast, spend a lot of time over there. They go to the Keys in the wintertime, and then occasionally, these 7 fish actually came over to the east coast and were detected over here. So at any one time, you could catch a sawfish from the east coast on the west coast, and you could catch a sawfish from the west coast on the east coast. Or you could go to the Keys and catch anything. So that's kind of the broad overview of what sawfish are doing when we catch them over here, but we wanted to dive into a couple of individual case studies to look at what they were doing. The first fish that we'll look at was a large, about 13-foot male that we caught in the St. Lucie on rod and reel. And unfortunately, when we caught this guy, he came up with a big bundle of about 200-pound monofilament line wrapped around his body. And you can see in that picture to the right, that monofilament line was actually so tight around its head that it was digging in several inches on either side. An interesting note about this is, I had mentioned earlier that the males are pretty rare, and they're especially rare in the summertime, but we actually caught this fish in August of 2023. We were pretty optimistic because, we cut all that line off of him, we cleaned this guy up, we put a 10-year acoustic tag in him, and we sent him on his way. And he stuck around the IRL for about a month or so before eventually making his way up the coast and being detected out by Cape Canaveral, down by Melbourne, and then making his way back towards the St. Lucie after September. And then, after a few months, he was detected down in the Keys for the first time in January of 2024, and he spent about a month swimming around down there in the Keys, being detected by some of the offshore wrecks and spending time there until February. Unfortunately, 2 weeks later, he was found dead at Long Key State Park, and he is just one of several sawfish that were affected by an unusual mortality event in early 2024 down in the lower Keys. But, luckily, this carcass was reported to us through our encounter hotline, so we were able to get our hands on this carcass and collect valuable samples from it. What's really a bummer to me is that you can see in this picture here to the left that those wounds that we saw in the previous picture, in just 6.5 months since we tagged him, they were almost fully healed, and you could just see those scars there. So he was doing really well for 6.5 months, and then he found his way into a pretty bad situation down there in the Keys. But luckily, we were able to necropsy this animal, and hopefully some of the samples that we got from him will help us to figure out what's going on and try to prevent this from happening to other animals as well. Another example, or a case study, is this large female that was entrained at the Hutchinson Island power plant. She was about the same size as that other male, and she was tagged in January of 2023. She's one of 4 sawfish that we've been able to get our hands on that have been entrained in the power plant. So when they tagged her, she pretty much immediately left the IRL, and she headed down to the Keys and Florida Bay, which is kind of what we would expect that time of year. Again, they seem to overwinter in the Keys. So she spent some time down there throughout February and March. And then by May, she actually moved up the West Coast and was detected off Tampa Bay and spent quite a bit of time up there. And then she did something really cool, at least in my opinion. She headed all the way up to Apalachicola Bay, which is the furthest north that any of our tagged sawfish or any of the fish that we've put tags in have gone on the Gulf Coast of Florida. But she then immediately headed back south, or a couple months later, and was detected for the first time in the Marquesas and the Lower Keys before making her way back up the East Coast. So she's very well traveled in a fairly short amount of time. And then, just like all the other sawfish, she seemed to overwinter in the Keys, and she was last heard from in March of 2024, which is terrible timing, but we're going to be optimistic that maybe this is just a delay in receiver downloads, and hopefully we'll get more data from her in the future, and she didn't meet the same fate as her friend. So in the future, we want to continue this work. We want to continue to get more data from the data sharing networks. We'd love to spread our sampling out over more of the year so we can see if some of these trends that we're seeing reign true. And we want to see if we continue to catch mostly males earlier in the year, mostly females later in the year. And we want to just continue to track these animals because the tags that we're putting out are 10-year acoustic tags. And most of those data you just saw are only maybe 2 or 3 years worth of data. So I want to just kind of finish that out. And we have lots and lots of questions to continue to investigate with these fish. So with that, thank you to our funding sources and all the data contributors, and I'll take any questions if we have time.
All right, thank you, Andrew. We have time for one quick question. Yeah, Lorraine?
It appears that the fish that you tagged on the West Coast started most of their big chunk of time over there and then they moved to the East Coast. Is that life history or environmental?
Probably. So weirdly, on the east coast of Florida, our sampling that we do over here, as I mentioned, we catch mostly adult sawfish. But on the west coast, we've actually, in 20 years, we've only caught 4 adults. So most of those fish that you were looking at were large juveniles or even kind of like medium size. And so, yes, it would take longer for them to maybe start that migration.
All right. Thanks, Andrew. Okay. Up next, we are going to hear from Ari about phycotoxin exposure in white-spotted eagle rays.
Hi everyone, thank you for being here today and listening to my talk. So I'm going to be telling you about the exposure of white-spotted eagle rays to phycotoxins in Florida coastal waters. So as most of you know, harvest alga blooms are expected to increase with climate change. However, we don't know a lot about how toxic HABs specifically affect marine wildlife. Specifically for groups of animals that I really like, which are sharks and rays, we have very little information about how toxic HABs and phycotoxins affect them. Sharks and rays are exposed to phycotoxins through their diet and also uptake across the gills. They're also susceptible to bioaccumulation and biomagnification as top predators and mesopredators, but also they have high lipid contents and slow growth. The white-spotted eagle ray here that you can see here, this species is affected by phycotoxins, by feeding on shellfish, also through bioturbation of sediment-bound phycotoxins, but also they rely on have impacted areas throughout ontogeny. However, this species has also been assessed for uptake of multiple phycotoxins. So why should we care about this? Well, these animals are really cool. That's the first. The second, this is a globally endangered species and it has a declining population trend. The other thing is that this species relies on potentially nursery habitats, which we are studying, that are the Sarasota Bay area and also the Indian River Lagoon. So one big question that we have is like, how are HABs affecting these rays in these locations, and how HABs can impact the little young, the pups that stay inside the nursery habitats. So our goals for this study are to evaluate phycotoxin exposure at wild populations of eagle rays in 2 potential nursery areas, and also evaluate the uptake by tissue and throughout ontogeny. So sample collection was in Sarasota Bay and Indian River Lagoon during long monitoring programs between 2018 and 2023. Samples that we collected for this study were blood and biopsy punches from the dorsal wing of the eagle ray, and then we collected gut content from via gastric lavage. We have liver samples too that we collected from euthanized rays and also deceased animals during red tide events. Samples from 34 rays from the Indian River Lagoon and 39 rays from Sarasota Bay were sent to the University of Connecticut to study, to determine phycotoxin concentration with UPL-CMS. And as you can see here, we have toxin concentration for 14 different toxins, which included domoic acid, brevetoxin, microcystins, and so many more. Here you have the pie charts representing the count of detections for each location. You have the Indian River Lagoon on the top, and you have Sarasota Bay on the bottom, with the tissues from left to right as plasma, muscle biopsy, gut content, and livers. And so you can see here there was a lot of diversity of phycotoxins in both locations, and several toxins were detected in both locations, and this included saxitoxin, domoic acid, microcystins, ocadic acid, teleocidin, and brevetoxin. In addition to this, other toxins, neosaxitoxin, cylindrospermosine, and nodularin were also detected in the Gulf Coast. So, and this resulted in a higher diversity of toxins for West Coast rays. So we think this is likely due to the migration patterns of the white-spotted eagle rays in the west coast of Florida. This population is highly migratory. They have a range that encompasses most of Florida coast and west coast, and they do repeated migrations between hab-impacted areas, such as Sarasota Bay, Charlotte Harbor, and potentially the Florida Keys, likely increasing their exposure to several habs. So this is a proportion of positive samples for phycotoxins by location, tissue, and life stage. So you can see the location up top, SP is for Sarasota Bay, and you will see IRL in a minute. And then you have in the x-axis, you have the tissue type, P, M, G, C, and L for plasma, muscle, gut content, and liver. And then you have here the different life stages. You have young at the year, similar to the sawfish. This includes like the newborns to 1-year-old rays, and you have juveniles and mature rays. As you can see here, in the plasma, brevetoxin was detected in all life stages at a higher proportion, and so this was kind of expected in the Gulf Coast, given the presence of Carina brevis in this location. As you can see here, we mapped the distribution of Carina brevis during the study period between 2018 and 2023 with overlap with our captured locations. As you can see here, this is evidence of the high exposure of this population to Carina brevis or to red tides. In addition to this, other toxins like cylindrospermosine in blue and microcystin in green, you see they were detected in the plasma in all life stages. In addition to this, we have evidence of trophic transfer of domoic acid and teal up there, here, and teleocidin on the gut content of immature rays, On the east coast of Florida, you can see here macrocystin in green and domoic acid in teal. They were the most abundant toxins detected in samples from the east coast of Florida, and this is likely due to the persistent presence of Pseudoniscia and Microcystis aeruginosa in the IRL, and also their toxins, domoic acid and microcystin, that remain in the environment for long periods of time. Domoic acid and microcystin can get into the sediments. They can also be accumulated by filter feeders, and they can be resurfaced to the water column, exposing these animals, which are benthic feeders and bioturbators, as you saw in the video, exposing them not only during blooms, but also long after a bloom has dissipated. In addition to this, you can see the highlight in this plot is that domoic acid and microcystin was detected in the gut content and the liver samples from young rays. These are newborn to 1-year-old rays, And so this is evidence of trophic transfer, right? Whatever they're eating has phytotoxins, and they're getting phytotoxins this way. And this is also a potential for accumulation of these toxins in little rays. As you can see here, the same thing happens for juveniles, which is kind of worrisome in the IRL. And so this is just to show you how these rays are repeatedly exposed to some of these toxins. As an example, in the Gulf Coast of Florida, you see here some plasma samples throughout the years from 2020 to 2023. You can see here our eagle rays were constantly exposed to brevetoxin in red, and they were also exposed to microcystin in green and cylindrospirmosin in blue 2 consecutive years. A similar pattern was observed in the Atlantic coast here in the IRL. This is where you're looking at is the gut content here throughout the years, and then you see here in the first 2 how rays were repeatedly exposed to microcystin these 2 years, and then how domoic acid was detected in 2018, 2020, and 2021. So this study provides evidence that eagle ray populations in Florida are repeatedly exposed to low levels of phytotoxins, raising questions about the long-term effects it has on their populations. This study also provided baseline concentrations for various phytotoxins for wild populations of rays and evidence exposure throughout ontogeny. It also showed dietary exposures as an important mechanism of transfer for this species, as well as that young eagle rays in the IRL are exposed to phytotoxins via trophic transfer. Thank you to my team and the funding sources and you all for listening to this talk.
All right, thanks, Ari. We do have time for one quick question. Everybody's so quiet after lunch. All right, we'll catch up with Ari if you want to learn more at the break. Okay, next we're going to hear from Justin, who's going to tell us about burrowing fauna in the IRL and show us some awesome pictures.
Let's take a break from all the data and graphs and look at some animals. All right, so hi everyone, I'm Justin. Thank you for coming. I'm a postdoc at the Smithsonian Marine Station in the Benthic Ecology Lab, and today I'm going to talk to you guys about an often overlooked and understudied component of coastal marine faunas, which are benthic associated marine invertebrates. So those of you guys who've spent some time in the lagoon and looked down have probably noticed things like this, which are macroinvertebrate burrows. They're pretty abundant, especially around the inlets of our lagoon, and come in a wide variety of shapes and sizes. And if you're a small invertebrate in the IRL, there are a number of reasons why constructing a burrow, living in a burrow might be adaptive for you. So first of all, you've got to deal with all these nasty fish and birds that are trying to eat you all the time, and it affords you some protection from that. Also, if you are living in an intertidal ecosystem, you're dealing with the tide coming in and out all the time, and with that comes rapid changes in temperature, potentially salinity, if you get exposed and so on. And so a burrow can act as a refuge from some of that environmental stress. And also it can potentially offer you a source of food, accessing organic material deposits or plant roots and things like that. But of course, it comes at some cost. First of all, you've got to spend the energy to dig it and maintain it, which is not easy. And you're competing for space against both conspecific and interspecific organisms that also want to build burrows. So burrows are a valuable resource, but also a pretty expensive resource. So what if I told you you could reap all the benefits of a burrow without having to spend the metabolic cost of building one? Well, a lot of things have this very idea. So there are a number of organisms across many different animal phyla that cohabit the burrows of other organisms. So there are crustaceans that do this, annelids that do this, flatworms that do this, and many others. Even some gobies, like the one pictured here, which acts as a watchdog for snapping shrimp that they live with as roommates. And so just like the cohabitants are diverse, they also occur with a diverse array of hosts. So there are things that live in crustacean burrows, things that live in annelid burrows, and so on. And these associations can be obligate, meaning that, you know, this symbiosis is a necessary part of this animal's life cycle, or facultative. So there are some things that will cohabit burrows when they're available, but also can occur as free living. And these symbioses run the gamut from mutualistic, like this one that I described a moment ago, to parasitic, but most of them are considered to be commensal, meaning not having any real noticeable fitness effect on the host, but for the most part, that's because we don't know enough about the interaction to say. And so because burrow cohabitants are often tiny, they're hard to get, they're hard to collect, these things are underground, underwater. So essentially, these are animals that if you're not looking for them, you're not going to see. And as a result, broad-scale sampling surveys often miss this component of the fauna entirely. So if we're interested in having a holistic picture of biodiversity, you know, difficult to collect specific faunas like these are things that have to be targeted. So what my project with SMS strive to do is to target burrowing animals specifically across the Indian River Lagoon. And so went out 39 times over the course of about a year from 23 to 24 and sampled in 14 sites across the 5 inlets of the IRL, as well as down at Peanut Island in the northern part of the Lake Worth Lagoon. And so sampling was always conducted during extreme low tides because that's when burrows are most accessible to get to with this device, which is called a yabby pump. It's essentially a giant stainless steel slurp gun. And so the way it works is you just suck up things from the aperture of the burrow and then quickly eject the contents into a sieve that hopefully you convince someone to hold for you, and you hope for the best. So then all those specimens were brought back to the laboratory to get photographed, to document their living color, took tissues for DNA barcoding, and preserved them for morphological identification. So in total, we got 607 critters doing this, and a quick and dirty molecular species delimitation identified 117 operative taxonomic units, which are essentially molecular species, right? So that's a lot. 23 of those 117 are new to our Indian River Lagoon species inventory, so they're new records for this region, at least 23. And 52 of those 117 had no existing DNA barcode data, so almost half. So this is, as I said, a part of the diversity that is not getting paid attention to. So when we look at it taxonomically, this is a pretty worm and crustacean-dominated fauna, right? Mostly arthropods and annelids, but there is also a cast of weird other characters thrown in there as well. And about half of them are things that construct their own burrows, 33 are known symbionts, and then the rest are potentially facultative associates or just essentially bycatch other things that are living in the surrounding sediments. So let's talk about what some of these characters are, because many of you may not be familiar with these organisms. So the most abundant burrowers in our lagoon are mud shrimps, so members of the infraorders Axiidia and Gabiidia. They are the ones who construct those big volcanoes that you might see sometimes. Snapping shrimps are also pretty abundant burrowers, as are capitellid worms or arenicolid worms. Mantis shrimps make some of the most distinct and large burrows, some of which can be meters long and have these kind of cool wide apertures. Also got things like acorn worms and mud crabs as well. So some of the cohabitant groups, the probably most diverse one are the pea crabs or pinotherids. Some of you may know these as endosymbionts of mollusks that live inside their mantle cavity, but there are actually a number of them that live in association with burrows. Several snapping shrimps also are obligate symbionts, and actually since starting this postdoc, I've described 2 new symbiotic snapping shrimps that occur in the IRL, so there are still species to be found right here in Florida. And another sort of interesting character to point out are the yo-yo clams, which get their name from hanging from the top of stomatopod burrows like a yo-yo, and when the mantis shrimp starts to aerate its burrow, they just sort of suspend themselves and filter feed some of the goodies in the water.
So I don't have time to talk about all the cool things we found, but I want to highlight a couple of them. One of which is this animal, Alpheus christophersoni. It's a symbiotic snapping shrimp that lives in the burrows of spoonworms in the genus Acetostoma. This is an animal that previously had only been reported from Panama and Colombia, so not only a new IRL record, but a new USA record. And it's a pretty rare animal, and so they don't often get seen. And what was cool to see about it is when putting it in a dish with its host, as the worm sort of locomotes around, you can see the symbiont follows it everywhere it goes. It doesn't really want to leave its side. So kind of an admirable fidelity there. But yeah. Another interesting find is these red platyhelminths, red flatworms that live in association with some mud shrimps. Found some of these in Peanut Island as well as around Fort Pierce Inlet. And I don't know what they are. I'm pretty sure they're undescribed. We've contacted some people who work on these things and compared sequences, and it's nothing like anything that's been documented before. So quickly, when looking at sort of the summary of who's living with who, we find some interesting patterns. So each of these boxes is a species, on the top row being burrowers and the bottom row being cohabitants. And when we compare this network to some other networks, what's interesting is looking at the pattern of nestedness. So quickly to say, nestedness is this pattern of sort of a linear relationship of generalists to specialists on both sides of the equation. So there's generalist to specialist cohabitants, generalist to specialist hosts. And so when comparing this to other networks, what's valuable about measuring nestedness is highly nested networks tend to be mutualistic, lowly nested networks tend to be more parasitic. And the N value, which is a sort of measure of nestedness, falls within things like reef fish cleaning, pollination, and seed dispersal, which are mutualistic. So while we don't have evidence that most of these interactions are mutualistic, it seems like the network resembles mutualistic networks. So moving forward, what I'm doing to kind of build on some of these results is, first of all, I built this ant farm. And we're doing some in-burrow observations of what the symbionts are doing in the burrows with those, and also creating resin casts to try and get some CT images of the burrows themselves. And with that, I've got people to thank, and I think that's time.
Thank you, Justin. We don't have any time for questions, but if you are interested in learning more, make sure you catch Justin at the break. Okay, we're going to move on and hear from Lauren, who's going to talk about Lake-O discharge and bottlenose dolphins.
Hi, I'm Lauren, and I'm part of the Marine Mammal Stranding and Population Assessment team here on Harbor Branch's campus. So for those of you who don't know, bottlenose dolphins are environmental sentinels, meaning that their health can be reflective of ecosystem health. They also exhibit site fidelity to varying degrees, where they will return to areas over and over again. And this is potentially problematic for those that exhibit high site fidelity because they don't necessarily leave areas that could be harmful to them. Typically, they're in habitats that range from 15 to 35 parts per thousand for salinity, but they've been documented above and below this threshold. Some of the effects of low salinity are that dolphins may avoid or leave areas with low salinity. They may develop these freshwater lesions. It can alter the electrolytes in their blood or in their eye fluid and other fluids in their body. And this can make them more prone to bloodborne illnesses. It can make them more susceptible to infections. And generally, freshwater exposure can cause death in dolphins. It is very hard to define thresholds for dolphins with this because dolphins not only can come and go between areas of low salinity and high salinity, but there's still a lot that we don't understand about freshwater effects. But what we do know is that the most problematic for them is prolonged exposure. But setting these boundaries of what is considered prolonged and at what salinities and how long it would take to recover is varying by many regions, and there's still a lot of research that needs to be done. Some thresholds that have been set vary from anything less than 15 parts per thousand to less than 10 parts per thousand or at 10, and also behavioral changes being noted at 5 parts per thousand of even complete avoidance of some areas. The St. Lucie estuary system is one of the systems that dolphins do inhabit, including the river and up through the North and the South Fork. And it's also prone to freshwater discharges. So for those of you who don't know, the South Fork is connected to Lake Okeechobee through the C-44 canal. And this canal is opened for flood relief much of the time. And when it brings this water with it, it can be water of low quality with algal blooms in it potentially, or algae in general, and also lower salinities. And all of this can be harmful to marine life, including dolphins. So our question was, in an area such as this, are dolphins' movements affected by these openings? So we used photo ID to monitor these animals. We do run Sebastian to Jupiter Inlet on a monthly basis. We follow along these sawtooth transects until we encounter a group of dolphins, and then we stop and collect data. For the sake of this study, we focused down just from South Hutchinson Island down to Jupiter Inlet, and then also into the St. Lucie River and North and South Fork. So to monitor dolphins, we do use dorsal fin images for identification, body images for lesions and wounds, life history information, and noting behaviors as well. And at the end of all of our sightings, we do take water parameters with a YSI, such as salinity, pH, conductivity, dissolved oxygen, and water temperature. So for flow data, to quantify how much flow was coming through and when, we used the DB Hydro through South Florida Water Management District at this station, S80, because it's what is directly emptying into the South Fork itself, and this flow is in units of cubic feet per second. And we use this data to also identify peak flow from 2022 to 2024. To monitor how salinity was changing with this flow in our study area, we pulled salinity data in parts per thousand from the Erlon stations at the North Fork, South Fork, Midpoint, and at the Crossroads. And then we calculated an average salinity for each of the months when the peak flow was happening. And if part of the month there was flow and part of the month there was not flow, they each got their own averages, so some months are going to have 2 points. So what we found was that for 2023 and 2024, which is noted by the colored bars here on the graph with month on the x-axis and flow on the y, there were these peaks of February through April. Whereas in 2022, which we included here more so as a reference year, there was little flow from January to May, and we left January and May on there as the study end caps. And interestingly enough, if you look at the purple bar for 2024, within February and March alone, some of this flow was more than was discharged within a whole year with historical data. So quite a bit of flow happening. As for these average salinities, again, if a month has 2 points, it means that some of the month there was flow and some of the month there was not. For 2022, so we have month on the x-axis and average salinity on the y. That top station there is the Crossroads station. The middle one down with the dark line is the mid station. The black line is North Fork, and that bottom light green line is the South Fork. And there you'll see that horizontal line at 15 parts per thousand, which is about that habitat that dolphins spend a lot of time in. So none of the salinities dipped below that point. For 2023, when the peak flow was between February and April, the salinities dip below that point a little bit, but it's nothing drastic. But moving into 2024, not only does it drop below that 15 parts per thousand, in some cases it drops below 10 and it drops below 5. So moving forward, we're only going to talk about 2024 for dolphin sightings, but 2023 and 2022 up here provide those excellent comparisons of how much flow and how much the salinity had changed. So for 2024, this is a map of dolphin sightings, and we surveyed in the area before flow had happened at all. So this is a pretty standard month for us in terms of photo ID sightings. And as we move into February, those purple dots, the area was surveyed before flow started. So we don't know what it was like during flow, but down those blue dots closer south to the Jupiter area, that's a run that we typically have little to no dolphin sightings in, and this month we had 4. So it does appear that dolphins are moving, could be moving out of an area and going down further south into Jupiter. In March, which was a month of whole flow, there were only 3 sightings total for this entire area, and there were a couple sightings in the forks at this time, and the salinity at that point was 10 or less parts per thousand. In April, there was no flow, and there's only 4 sightings total. Again, 1 sighting in the Forks area, but that was back up to 15 parts per thousand. And in May, the number of sightings start to increase again, and this is with 1 month or more of no flow. So from our study, it appeared that there were less sightings and that there could have been some displacement into other areas, potentially even out of the map range that we showed. They could have used that south route and gone farther north, and this potentially could have been due to the changes in salinity, but we would have to have additional years of data to really see if this trend holds with more spillway openings. The survey is also not designed around spillway openings. So we don't, for months that have some flow and no flow, we don't have surveys before, during, and after every single one. And we did not account for group size, so potentially if we were seeing a lower number of sightings, it could have been because the dolphin groups were larger, and we didn't account for annual variability. However, despite this, what we did find was that some of the dolphins were sighted in areas of low salinity at 10 or less that could potentially be harmful to their health, and this could also be said for other marine creatures in that area. And we hope that it contributes to the understanding of freshwater exposure, which is limited at this time. In the future, we would like to go back to when the spillway has been opened and monitor for any kind of skin lesions that could be developing in conjunction with that freshwater exposure, and then continue to map movement of dolphins in the future as that spillway is opened. And finally, it would be interesting to link this to presence and abundance of prey items. So if those dolphins were displaced down to that Jupiter area, were they following food? Or for the ones that did stay in the Forks despite that really low salinity, was that because they still had food sources there? And with that, I would like to thank all the following people and the FAU and Harbor Branch Foundations and all the staff past and present, including Tracy Kowalczyk and Wendy Marks. Thank you.
Okay, thanks, Lauren. We do have time for one quick question. Anybody? Yes.
I'm sorry? No, I didn't. So rainfall definitely could have played a part with just that natural freshwater input that comes into that system. But the salinities in general really didn't dip below that 15 parts per thousand line, even when there was rainfall. So it seems that at least for those couple of years, the spillway openings were what really could have potentially caused that dramatic drop in salinity.
All right, thanks, Lauren. All right, to round out this session, we are going to now hear from Sarah, who's going to talk to us again about sawfish.
These are the more important ones, the little ones that Andrew didn't want to talk about. So good afternoon. I'll try to go quick to not keep you guys from the pizza party, but today I'll be discussing the movement and distribution patterns of endangered small-toothed sawfish in the recovering nursery. So a little bit of background information. The small-toothed sawfish is one of 5 species found worldwide, but the only one found in Florida, and its historical range used to extend from Texas through the mid-Atlantic, but most occurrences now are within southwest Florida. This fish was federally listed in 2003, and its decline was due in part largely to entanglement in fishing gear and loss of essential habitat for those younger life stages, such as red mangroves. A critical juvenile habitat was designated by NOAA in the Charlotte Harbor Estuary and the 10,000 Islands and Everglades unit. There had been little to no research on this ray before it was listed in 2003, but in Charlotte Harbor area, the small juveniles are known to be born in the rivers and mainly stay there in shallow waters. And there's well-documented nurseries and known juvenile hotspots in both the Peace River and Caloosahatchee River. However, sawfish in the Indian River Lagoon used to be an abundant and permanent resident, with examples often around the 3 foot in total length, so again, back to those small juveniles, so that may be indicative of this area potentially serving as a nursery at some point. And one fisherman reported catching up to 300 fish in a single season. Gilmore and Snelson and Williams actually documented this fish as being extirpated from the area by the 70s. However, small-toothed sawfish recovery plan did identify that the IRL was important for the recovery of this species, and Waters et al. Suggested a resident population offshore of the area, with larger juveniles and adults being observed. So Harbor Branch fits into this too because the Fisheries Ecology and Conservation Lab, run by my advisor, Dr. Matthew Ajamian, actually captured one of the first adult sawfish in 2016. They also were responsible for catching one of the first small juveniles in 2020 and worked with FWC to collect another small juvenile and tag it in 2020, as well as continued collaborative work at the power plant. So for the purposes of this study, again, the important sawfish, the little guys, we're not going to talk about the big ones. Andrew already had his time. So again, like these freshly born, the young of year, and up to the age 1, so not really anything over 5 feet. The overall purpose of this study is to identify the status of the St. Lucie estuary as the nursery habitat for these fish and examine the movement and residency patterns using acoustic telemetry and identify hotspots through acoustic telemetry movements, as well as utilizing the encounter hotline data to look at where encounters occur most often in the estuary. So looking at this, we could see that there was a notable increase in encounter reports in the St. Lucie area, especially from 2019 to 2023. In the green dots, you'll see the small juveniles are about 109 total encounters, but it went from 2 in 2019 to over 30 in 2023. So this could be due in part because people are more aware of the hotline now. Dr. Polakis will talk about that a little bit more, or maybe there just really are that many more small juveniles around. So using that encounter data, a density mean cluster analysis was created to identify clusters of high encounters. So the kernel density estimation on the left-hand side of your screen, got to figure out which way I'm turned, is the kernel density estimation with the higher encounters in that bright purple that you'll see kind of in the South Fork area. And then 3 of those mean density clusters were in the South Fork, 3 of the 8 that were totally populated. And those were essentially around like fishing piers and bridges. This is a map of the study site with the capture locations of the tagged juvenile sawfish in the circles, the acoustic telemetry stationary receivers noted as triangles, and the Erlon stations in the squares with the dots in them. For the purposes of this presentation and for this part of the study, we utilized Erlon information from the South Fork, Middle Estuary, and North Fork. We also obtained flow data through the South Florida Water Management District DB Hydro. On the far right, you'll see a list of the tagged individuals and where they were located. On the bottom are fish that were too small to hold an acoustic tag, so they were pit tagged, kind of like a microchip you put in your pet for later identification in the event of a recapture. You'll also notice 2 sets of the same date, so 2 fish were actually caught in the same net on the same day, and later were, through genetics, identified as full broodmates, so that was pretty cool. And then the other set that was pit tagged found out that they were full broodmates because we were there. So this is always fun, something I never thought I'd be part of, so it's really cool to share this with everybody else as well. So continuing from that, from the acoustic data, we created a kernel density utilization estimate, and you'll see in the South Fork is where some of the sawfish are really spending a lot of their time. And there's only 2 stations in the South Fork. There's only 2 acoustic stations where these fish could be detected at, and yet they're being detected there significantly more than anywhere else in the estuary that they are being detected. During this study, in that size class, none of these fish were detected outside of the Middle Estuary, is why we didn't use the Erlon information near the crossroads area. And then for general additive models, we looked at flow, mean dissolved oxygen, mean salinity, and mean temperature during the study period. And there was a significant effect of flow, dissolved oxygen, salinity, and temperature on the fish location during this time in the study. With an effect with salinity, when salinities were low, there was a negative effect and fish were moving downriver. And so here's an example of one fish with 0 being the inlet, and then the salinity in the blue squiggly line. The location throughout is the black dots over the course of the time that this fish was considered still a small juvenile. So we'll watch him do a little dance, going through. And so as salinities went back to a more favorable conditions for these fish, the fish would move back upriver towards the South Fork or Middle Estuary area. So in conclusion, the South Fork is a really important area. The residency indices revealed that juveniles spent up to 90% of their detection days in the South Fork, supporting its role as a critical habitat. And there was also a significant influence of salinity, temperature, dissolved oxygen, and flow on the habitat use of these fish. And movements within the estuary were seasonally influenced by the freshwater inflows, with juveniles relocating downstream during those low salinity events. And so protecting and restoring the high-use regions, such as the South Fork, is essential for supporting population recovery and enhancing the resilience of small-tooth sawfish across their historical range. So Dr. Polakis, again, is going to give a shout-out about this, but the hotline's been integral in this. We've actually been able to go out like the next day and relocate fish that were called in from the hotline and be able to tag them to track them for this study and for the future.
And with that, I would like to thank my co-authors as well as a number of collaborators,
I don't even have to
and
say
supporters.
I didn't know that was on there, so I'm sorry. Here are my references. And if you have any questions, let me know.
Thank you, Sarah. We do have time for a couple questions. And I
might
think
be
that's
able to record
Nicholas?
the answer, too.
Yeah.
In previous publication, okay, so the question was, what is the salinity range for the small-tooth sawfish? In past publications, it has been the 18 to 30 range, and the same with the general additive models, and this kind of revealed the same thing, that in this area, too, they prefer that, so they'll move downriver to where it's a little more saline, and then back upriver when it becomes a little more stable again, into that range.
Jim?
Without seine nets, what's their biggest threat? Pollution probably still is one of them, and then urbanization, I would say, loss of habitat.
Okay, all the way over there on the, on the side.
What would my recommendation be policy-wise or conservation-wise moving forward, knowing that the South Fork is an important area for this species? I would say just further habitat restoration and just maybe better salinity regimes.
All right, thanks, Sarah.
Thank you.
Okay, before we move on, before I hand it off to Kelly for the 3 minutes, before I hand it off to Rachel for the 3 minutes, 3 slides, I neglected to mention that Sarah and our first 3 presenters were student presentations. So let's just give them and all of our presenters one last round of applause.
All right, so we're moving into the next 3 minutes, 3 slides. I am filling in for Kelly for this. This is on fish and food webs. If you are presenting in this and you're not sitting in one of these front 2 rows and you'd like to move a little closer, please feel free to do so now. So the first talk will be by Arthur Jones. There it is. A regime shift is reflected in the fish community structure of the Indian River Lagoon. And I'll be staying up here during the talks, I should say that. I'm going to start your timer right now, and at 3 minutes, Kristen is armed with the net. If you are not done, she will get you. So I'll let you take your place.
How do I,
right and left. Okay. All right, and then your timer's going to be here. You ready? 1, 2, go.
Today I'm going to be talking about fish communities and regime shifts. In 2021, Flipser posed the hypothesis that the IRL underwent a regime shift from a seagrass-dominated community to a phytoplankton-dominated one, and that this regime shift was caused by the 2011 HAB superbloom. As you can see by this graph, which shows phytoplankton biomass over time, phytoplankton maintains a low, stable, steady state up until 2011, when there is a massive peak. And then for many years after that, there continue to be massive peaks. Conversely, as you can see in this graph of seagrass coverage over time, seagrass maintains a high, healthy, stable state up until 2011, where there is a sharp decline. With this regime shift in mind, we wanted to answer the question, is this regime shift reflected in the fish community dynamics, including mass mortality events in the IRL? we'd like to thank the FWC for providing data for this research. We first looked at fish kill information from the past 30 years. As you can see from this graph of total fish mortality over time, there is a massive peak in 2011 when the superbloom occurred and the regime shift occurred, as well as another one in 2016, both of which coincide with major superbloom events. In addition, you can see, as you can see from this graph of fish kill events over time, which are color-coded based on the suspected cause, you can see that the proportion of fish kills that can be attributed to algae blooms, color-coded in red, increased notably after the regime shift. Next, we looked at fish abundance over time, specifically juvenile and adult pinfish and sea trout abundance over time. As you can see, from 2010 to 2012 in all of these populations, there was a severe decline in abundance. This may be an indication this decline in abundance is driven by these HAB events, and which cause both direct increased mortality through fish kills, as well as a loss of seagrass habitat, which is their nursery habitat. What are our next steps for this research? We'd like to look at the population trend graphs for 8 more species of interest. We would like to make a map of fish kill hotspots in the IRL. We would like to conduct change point analysis on the population trend data and fish mortality data to see if the change from before and after 2011 is statistically significant. And lastly, we'd like to understand how this research contributes to our understanding of how HAB events affect the health of ecosystems like the IRL as a whole, and emphasize the importance of restoration and sustainability efforts, especially seagrass restoration efforts. Thank you.
All right, great, thank you. And that was a student presentation also, just for your voting needs. The next presentation will be Eric Alt, talking about offshore snook in Southeast Florida. And this is what Kristen was for, sorry.
So I just get extra time, that's how that
Apparently,
works,
yeah. All right. Go.
All right, guys. So I wanted to start off with a little bit of background information. So I'm part of a marine fisheries team out at Aquesta, and in the late 90s, we had a project that was looking at snappers and groupers, involved a lot of offshore diving and stuff like that. And during those dives, from time to time, we would encounter snook, sometime in large numbers. And that's, for us, it's mainly an estuarine fish, and you're seeing them out on coral reefs. We, kind of scientific curiosity, are they out there all the time? So we reached out to some folks in the fishing and diving community, some old time folks that had been around a while, had a lot of historic information. And they said, they came back with things like, yeah, we've seen them out there, but it does seem like there's been an uptick here recently. And some of the theories they put forth included a lot of things, including loss of habitat due from either natural or anthropogenic impacts on nearshore areas. And for us, it was like, does this represent a satellite population of the, of the general population? So we were interested. But it was something we tabled because it wasn't, we weren't focusing on that until 2008 when we embarked on a comprehensive snook project, and it was focused on the East Coast from Fort Pierce down to Jupiter, and we were looking at movement, spawning frequency, habitat utilization, and connectivity or exchange between the different habitat systems, which included this coastal offshore reef area. And the tools we had were acoustic telemetry for movement, specimen collections for life history, and underwater observations for documenting and describing things. And I'm going to talk to you about these underwater observations. And why we refer to them as observations instead of surveys, even though we utilize roving point counts and survey techniques, is that they were really biased in that we focused on an individual species. They were complementary, meaning we tacked them onto other things, and they were designed to give us baseline information, such as group size, number of individuals, the length frequencies, what's the general size class out there, their spatial distribution, and if there was a seasonal component to this. So, after 6 years of doing this, what we found is that when we did encounter fish, the average group size or mean group size was 23 and a half, even a half fish, right? So 23 and a half fish. We did see groups that were over 200. In terms of lake frequencies, because these are observations, I thought it'd be easier to give you a range. So most of the fish, over 70%, fell between that 750 to 949 total length in millimeters. And in terms of spatial distribution, we tended to see more in the St. Lucie and Fort Pierce, slightly more in St. Lucie than we did in Jupiter. But a cool thing is that we did see, while there was some fluctuation in terms of year-round, in terms of them being offshore, the seasonality was insignificant. So they're offshore year round. So from this portion of the information, we found evidence to support that this is a satellite population out there. And then the next steps were to try to build off of that. So with that, thank you guys for your time. And I'm sure you guys can hit me up and we can go from there. Thank you.
All right, next we have Greg Polakis, who's going to talk about sawfish recovery. And I'm going to just grab the other one because I'm not good at this.
All right, thank you. Just wanted to take a couple minutes. Sarah and Andrew did a great job, kind of talking about the hotline already. I added a please there for you guys. I mean, you guys are out on the water all the time. You talk to people that are on the water all the time. Please call us if you hear about sawfish or if you see them. Don't necessarily just encourage other people to call. You can call us. Second, third-hand information is fine. We'll be able to figure out if there's double reports and all that stuff. But for, hotline-wise, we get about 500 reports or so a year from the, usually Florida, mostly Florida. And most of that stuff is from Southwest Florida and the Keys, so this part of the world our state for sure, we're concerned about and have our finger on the pulse. We want to keep our finger on the pulse there. So, you notice in the graph here, a nice uptick that Sarah mentioned. This is all sizes combined, but 2022, 2023, about 30 or so, plus or minus 30, were those small fish that we've been talking about. But in 2024, there's only 9 of those 46 were small ones. So is that really what's going on, or have we just not been getting the word out enough? So like I say, we're just trying to make sure people are reporting, and we want to make sure that that's figure out if that's real or just an artifact of the reporting system. We try to get out and do as much as we can, but like I say, you guys are out and talk to the people as much as possible. So please let us know. We're answering the phone, we're answering emails. Those reports come in. If they come in real time, we will try to tag them. With Matt Ajamian's group and Eric Alt's group, they're all super interested in finding out about this species as well. So we will mobilize. We'll drive over from Port Charlotte even, try and get some tags out and kind of continue the stories that Sarah and Andrew were talking about today. So keep us in mind. Please let us know if you see them or hear about them. Thank you.
Thank you, with plenty of time to spare too. So next up we have Iris, who will be talking about environmental changes in bottlenose dolphins.
I wrote down, right?
Yes, to the right.
Yeah. All right, so accurate knowledge of animal diet, it's very important to understand predator ecology and evolution, to learn about the roles of predators in food webs as well as their foraging strategies. From the only study conducted in the Indian River Lagoon on dolphin diet, we know that seagrass meadows are a very important foraging ground for dolphins. They feed primarily on cyanid fishes and sonorous fishes using passive listening to locate their prey as a main foraging strategy. But they also feed on very highly abundant animals and very highly available, but all the common thing about all these species is that they are all seagrass-associated species. So what happened with this main foraging ground is lost. So we know like a combination of compound effect of changing climate and anthropogenic stressors are decimating the seagrasses in the Indian River Lagoon. So this is impacting, having a negative impact on fish populations, dolphin prey. How dolphins are coping with the loss of prey availability or the decline of prey availability. So to learn more about the IRL dolphin diet, in the last decade, we examined stomach content from samples from stranded dolphins that were opportunistically collected by SeaWorld, Hub SeaWorld Research Institute. So we, using a combination of direct stomach content and a few molecular techniques, metabarcoding metabarcoding and direct DNA barcoding, we are learning about what kind of prey items these dolphins are eating. So we still finding the Gulf toadfish and snappers are favorite for dolphins, and also some of the most common species. But like what I really want to highlight in this talk is, like, to my knowledge, and never been reported as a prey of dolphins, is the presence of pipefish. We have several cases where we find pipefishes and eels. There are some puffers too. So what these findings are telling us is, like, dolphins are not preying in their favorite food anymore, the big cyanide fatty animals. They are preying in lower trophic levels, likely with lower calorie content. They are also preying more creative species, which also increase the demand of energy for foraging, capturing, and handling their prey. And that results in poor body condition, and finally, emaciation, what we find in 76 of the cases. So yes, the environmental changes, the loss of the foundation species, seagrasses, are negatively affecting dolphin diets in the Indian River Lagoon.
All right, next we have Jessica, who will be talking about bridging the genetic gap to use DNA markers to assess the potential for meta-barcoding to infer ecosystem health in a subtropical estuary or a coastal lagoon.
Sorry, long title, long last name, so it works.
Oh no, I've ruined everything.
It's okay.
We'll
I can just go off of that. Can everybody hear me? Awesome, thank you. So my name is Jessica Zenfenig, and I'm a postdoctoral research fellow at the Smithsonian Marine Station. And today I'm just going to give a little overview of what I've been researching the past few months. So as many of you know, the Comprehensive Everglades Restoration Project, or SERP, has been funding quarterly monitoring of benthic and faunal species in the Indian River Lagoon. We monitor 15 sites along the St. Lucie estuary as well as up the lagoon. And one of the tools we use worldwide for this monitor is something called the MAMB index, or Multivariate At-Sea Marine Biotic Index. And that's an index that's been used worldwide to assess benthic health conditions. It looks at benthic in fauna and uses count data traditionally of these in fauna to give you kind of a report card score. And this is traditionally pretty expensive and time consuming. It relies on count data, and that count data has to be looked at. So we have to take a sample, look at the sample, sort everything in the sample, and then taxonomically identify it. This being said, environmental DNA is eDNA that has been found in the environment, so from a scoop of soil or water, that kind of gives you an insight of what animals have been there due to the cells left behind by those animals. This is a promising alternative to this kind of monitoring, as it's faster, cheaper, and can also often give more precise species level identifications. While this is promising, there's still a lot of work to be done in the field, including a limited knowledge of eDNA markers and reference libraries. So what we wanted to do is assess availability of these eDNA markers for ambi species found in the Indian River Lagoon. To do this, I looked at 5 common eDNA markers, CO1, 12S, 16S, 28S, and 18S, and I made a species list using the Indian River Lagoon species inventory and the 19 years of long-term monitoring data from the Benthic Ecology Lab at the Smithsonian Marine Station. From there, I cross-referenced that list against the most recent ambi species lists, and I took those species and looked against publicly available databases for those 5 markers. And since I had almost 1,200 species in the list, I sorted them into major taxonomic groups. There's 10 of them. And just really quickly, what I want to highlight is this donut chart gives the number of markers. So in this donut chart, the colors represent the number of markers that were available, 0 to 5, and then those smaller little charts are those major taxonomic groups. And 53.91% of the species on this list have less than 2 markers available, and only about 6.17% actually have all 5 markers. This is kind of an issue when you're trying to use eDNA to draw these conclusions. So one of the future works we're doing is we're holding a BioBlitz in June of 2025 to identify and barcode those infaunal species on the ambi list in the Indian River Lagoon. This will help eliminate those gaps, identify dark taxa, and then I'm also analyzing eDNA samples that we've taken along with the benthic monitoring to compare them against traditional sampling methods, and that'll help me investigate some real-world data. Thanks to everybody on this list, and I think I'm out of time, so.
All right, thank you, Jessica. Next up is Jim Whittington talking about movement patterns of Cobia snapper along the southeast coast of Florida. And there's your time.
Thank you. This cane is not for me. It's just to protect me from that net that's been Anyways, we're going to talk about the movement of Cobia snapper. Basically, these are subadults that are found in the Jupiter Inlet, Loxahatchee River area. We were fortunate to have recreational anglers and guides to help us capture these fish, and then they would call us, and then of course we would put an acoustic transmitter in it, and typically or standard procedures are to put it upside down, cover the head and gills with water, and make a small incision offset the midline and insert an acoustic transmitter. And that's what's shown here. And then these three over here are also dark tags so that if someone catches it, they know that there's an acoustic transmitter in the fish. And then that far picture on the right is a station where we are just taking the receiver and the data and swapping it out with a new one. So this is our preliminary results. We see that these fish have moved from Jupiter Inlet all the way up to Cape Canaveral and all the way down into the Keys. In fact, they've been found at Rowley's Hump and Western Dry Rocks, which are known spawning sites for other snapper species. And when you look at these, at the resident index of the Cobia snapper, you can see on the left side of the graphs that Jupiter, Loxahatchee, and Jupiter Offshore are high levels, but noticeably these areas to the south, these are south of Palm Beach, also have some fish that have spent some time there. So these are some of the guides that have helped us with the Cobia snapper. For some reason, they're holding snook and redfish. But anyways, we do like to give a shout out to the West Palm Beach Fishing Club for buying us the transmitters and also some of the receivers for the stations. Thank you.
All right, thank you. Next up is Jody, who's going to be talking about assessing trophic pathways of the southern Indian River Lagoon.
All right, so just jumping right into it, this study aimed to use fatty acids as trophic markers for harmful algae and their toxins. Fatty acids can be split into three different categories: polyunsaturated, monounsaturated, and saturated. We're going to focus a little bit more on the polyunsaturated fatty acids, or PUFAs, as I like to call them. They're typically produced by primary producers, such as phytoplankton and algae, and they exhibit conservative relative abundance throughout the food web. They also include essential fatty acids, specifically DHA and EPA, which have been positively correlated with high amounts of diatoms in the diet. One of those diatoms that has been mentioned before is Pseudonychia, a diatom species that's found in the IRL that can produce domoic acid, which is a potent neurotoxin and can also be a harmful algal bloom. It has a specific fatty acid marker, the 16,4N1 marker. We sampled 5 locations in the southern half of the Indian River Lagoon, from Sebastian all the way down to the St. Lucie Inlet. This culminated into this giant graph that's making you all blush. So we sampled 2 different trophic levels, zooplankton and fish. Specifically with the fish, we looked at dolphin prey species, so that we could be able to kind of infer that it would go all the way up the food chain. This was over the course of a year for both the zooplankton and fish. The big things to point out, we see the C16 marker, which is going to be that big yellow bar at the bottom there. That kind of indicates, that's usually pretty high for fish in estuaries, but is a little bit higher than normal, which can go to the zooplankton, we see the C16 fatty acid higher in freshwater algal species like Microcystis. So we can kind of infer a little bit of transfer there. We can also see the byproducts in the MUFAs, those two other bars, but the most important are the PUFAs at the top. We see this one I highlighted in red that has a couple little spikes. That's the pseudo-Nietzschea marker. That's going to be more spikes in November and February, which aligns with bloom time. And the two at the top, those are going to be the DHA and the EPA, which are, as I said before, highly associated with diatoms and harmful algal blooms. So we can see the very top bar and the third bar down are conservative between zooplankton and fish, which is important in being able to see the transfer of toxins throughout the food web and can be great indicator in the future of more frequent harmful algal blooms. Thank you.
All right, and just to note that Jody's presentation was also a student presentation if you're thinking about who you're going to vote for. So our last speaker in this session is Wendy, who will be talking about evaluating survival of entangled, free-swimming common bottlenose dolphins in the Indian River Lagoon.
Thank you, guys. Fishing gear is a growing threat to marine mammals, and in fact, it is one of the leading causes of mortality for dolphins residing in the northern Indian River Lagoon, specifically in our study site, which is from Sebastian Inlet North. Chronic entanglement can lead to severe injuries, amputation, infection, sepsis, and mortality, and is a particular threat to young animals that are going to continue to grow into the entanglement. In order to evaluate entanglement cases, we compiled free-swimming bottlenose dolphins from 2008 to 2023 in our study area. We used post-release monitoring to evaluate the success of our rescues, and using that success being defined as survival being greater than 12 weeks. Overall, we had 29 life-threatening entanglements that occurred in our area, and 75% of these were calves. 9 of these animals actually perished or shed the gear prior to intervention, leading 20 rescue efforts. So there were 14 net sets, 2 that we disentangled with remote tools, and 4 that were manually disentangled. Interventions typically occurred 26 days after discovery, and the majority of the entanglements consisted of monofilament or multifilament fishing line, although 10% were attributed to the crab pot fishery and 10% to other fisheries. Overall, 70% of our efforts were successful, although we did recover 20% of them deceased and another 10 were presumed deceased. Of the 6 unsuccessful efforts, all of these were calves, and 3 of them were so badly debilitated that they enabled a manual disentanglement. All of these animals were malnourished, had severe lacerations, a body wrap, or a constricted appendage. Ecosystem degradation within the Indian River Lagoon has corresponded with a 2-fold increase in human interactions and reduced nutritional health in dolphins. Therefore, increased entanglement risk may be due to ecological changes and decreased health, fueling risky interactions between dolphins and recreational fishers, or may simply represent an overlap between key foraging habitats and recreational fishing, or alternatively, just the accumulation of derelict gear. Mitigating these increasing impacts will depend on our increased capacity for quick response and a thorough assessment. We've found remote disentanglement can definitely expedite response efforts, and UAS technology provides a more complete assessment of both the severity and configuration. Ultimately, we're going to need to have continued targeted outreach to foster community engagement and to improve reporting. We would like to thank our funding sources, our collaborators, and thank you very much.
All right, thank you, everyone. That concludes that 3-minute, 3-slide session and begins our 20-minute break. There has been pizza delivered to the gallery. If you're hungry, please help yourself. There's pepperoni, lasagna, and cheese pizzas, so see what you want. Our next session is HABs and IRL restoration.
Hello. Hello. All right, we're going to go ahead and get started. First up, we have Jackie, who's going to talk to us about interactions of bacteria with pyrodinium.
All right, hello everyone. So today I'm going to be talking about Pyrodinium bahamense, and it blooms in the northern Indian River Lagoon. Here you can see a nice image of one of those blooms and the cell right here. And it's a urohaline thecate bioluminescent dinoflagellate, and it produces the potent neurotoxin saxitoxin, which can cause paralytic shellfish and pufferfish poisoning and result in human death. These blooms have also been implicated in mortality of fish, marine mammals, and birds, can attenuate light from seagrasses, and so obviously they have diverse consequences for human health, aquaculture, tourism, and ecosystem function. And so we want to know why these blooms are occurring and what factors are involved in these blooms. And we're finding that bacteria are really strong influencers of phytoplankton physiology. So how they exert these effects are through these small-scale interactions. So for example, they can have beneficial interactions. So many toxic dinos are oxytrophic or can't produce their own vitamin B12. And so bacteria like Rugeria can produce vitamin B12 for them. They also have detrimental effects. So some of these bacteria are found to produce algicides, which lyse and kill these cells. And so I hope with this you can see that these small-scale interactions can have large-scale implications. So beneficial interactions can probably help these blooms occur and persist, and then harmful interactions might be the reason why they collapse so well. These also are really important just for ecosystem function, how nutrients are recycled and moved up the food web, carbon cycling, and others, and how climate change might affect these communities and be one of the reasons why we're seeing so many recurring blooms of these harmful dinos. And so currently, we don't know much about the bacteria associated with pyrodinium in the IRL, and so that was the goal of this work. So first, we did some whole water sampling in regions in the IRL where we know that pyrodinium blooms. We took these samples and then did 16S amplicon metagenomic sequencing. So basically, bacteria have a prokaryotic ribosome that has a lot of homology in variable regions. So if we sequence this, we can look at the different bacteria that are in a mixed sample. We then wanted to look at those bacteria that are specific to pyrodinium. So we took single cells from these water samples and then made what are called unialgal cultures in the lab. And presumably when we add these single cells into nutrient medium, those bacteria will come along and be established with them in these cultures. And so we did, again, 16S sequencing on these as well. And then finally, we took these cultures and then plated them out onto nutrient agar to try to isolate these different bacteria that are culturable, and did whole genome sequencing so we can try to look at the genomes and see what kind of interactions they can be having with pyrodinium in the IRL. And so here's our sample collection. We have a lot of samples that were in the Banana River because those are that's where we're seeing a lot of these recurrent blooms. But we have some in the Mosquito Lagoon. And so I was able to collect about 7 to 8 isolates per site. And additionally, we had 3 isolates that have been kept meticulously since 2017 by Dr. McFarland. And additionally, we have water samples we collected as part of the Flora Center for Coastal and Human Health Initiative. And so here are the results of our 16S analysis for our co-cultures. And what you can see here is that there's a core taxa of phytoplankton-associated bacteria with the classes alpha proteobacteria in the light blue, gamma proteobacteria in the dark green, and Bacteroidea in the light green. And this has been found for other phytoplankton as well. When we look at the genus level of these different isolates, we found that only one bacteria was actually found in every single one of these isolates, and that was Neowella. And this is interesting because Neowella is found in the Rhodobacteriaceae family, and this clade, or this group of bacteria, is found in frequent and close association with toxic dinoflagellates, so that's consistent with pyrodinium as well. We also found that our isolates were predominated by several bacteria, which was interesting. So then when we looked at our water samples, we were looking for patterns there as well. And so here you can see the location of the water sample, the date collected, the pyrodinium cell counts, and then the relative abundance of Neowella. And what you'll see is that pyrodinium normally comes around the July months or the summer months in the wet season and then goes away during the dry season. And Neowella seems to follow the same trend. And so it seems that Neowella is mostly present when pyrodinium is present. So there could be a relationship there, but we need to do some more correlation studies to really investigate that. So the third thing we did, as I told you, is we tried to culture some of these bacteria. So we were able to get 17 unique bacteria cultured from our pyrodinium isolates and did some whole genome sequencing. And through that whole genome sequencing, we can start to model some of these putative relationships or interactions with pyrodinium. Obviously, this isn't to scale. The bacteria is much smaller than pyrodinium. But you can see some of the things that the bacteria might do in its relationships, such as it has an adhesive structure, which can help it attach to pyrodinium, a flagella to help it maintain spatial proximity. It can also potentially produce antibiotics, which can then compete with other bacteria for space around pyrodinium, as well as others like vitamins that could be really essential for its growth. So once we were able to figure out the composition and diversity of bacteria that are associated with pyrodinium, the next thing we wanted to do is some growth trials to see how these bacteria actually affect the physiology. And so we took some of our cultures and we made them axenic, which means we removed all the bacteria with a cocktail of antibiotics. So when we looked at the growth of our bacteria-free versus our xenic parent community, we found that the axenic culture actually grew better than the culture containing that parent community of bacteria. So that's a negative relationship we're seeing here. Then we can start adding single bacteria into that axenic culture and seeing how single bacteria have relationships. So here we see like a neutral relationship, so there was no change in growth. And then finally, for Alteromonas, when we added this single bacteria, it actually enhanced the growth of pyrodinium. We also looked a little bit into the toxin production changes that these bacteria can cause, and we didn't really see any significant effects yet, but we're still investigating this now. And so here I want to conclude, this is one of the first characterizations of the composition and diversity of bacteria associated with pyrodinium in the IRL. They were saying similar bacteria compared to toxic dinos in the literature. We found our regular associate is Neowella. We also found evidence of negative, neutral, and positive phytoplankton bacterial interactions. We don't have any evidence yet about how bacteria might impact toxin production, but we're still investigating. And so overall, determining this baseline information can help us discover how changes in these bacteria community, their metabolism and chemistry can affect pyrodinium physiology, how they bloom, magnify, and collapse. And then additionally, how climate change might fit into this scenario, how these changes and anthropogenic changes might affect these communities, and thus the dynamics of pyrodinium blooms. And so hopefully we can use this data to do some bloom detection, management, and prevention strategies as well in the future. And so with that, I will take questions, and if you want any more information, it's part of my defense, which is published. So thank you.
Since we're running a bit behind, we're going to let Jackie off the hook, but I'm sure if you ply her with alcohol tonight, she'll answer all your questions. Our next presentation is a student presentation. Manish is going to talk to us about sediment transport in eastern oyster reefs. Oh.
Hello everyone, I'm Manish Tenora. I'm a third year PhD student at UCF Ecohydrotic Labs. So today my presentation is sediment transport in eastern oyster reefs, specifically focusing on their turbulence and canopy density. So as we all aware, the oysters provide varieties of ecosystem services for the environment in terms of habitat creation, improving water quality, and most importantly, attenuating waves. And numerous research studies has been done in the past focusing on the hydrodynamic properties of the oyster reef. But very few research studies has been done focusing on the sediment transport aspect of these biological canopies. So in my research study, I'm mainly focusing on the investigating the sediment transport aspect of this biological canopy with related to the variant canopy density types. And also at the end of this research, I'm also planning to develop a framework to provide a better understanding about the sediment behavior happening within these canopies. So in order to achieve these objectives, I happen to conduct an experimental study. For that, I have selected a natural oyster reef located in Mosquito Lagoon. And within this, within this oyster reef, I have identified 4 different canopy density types, such as sparse, moderately dense, clusters dense, and the homogeneous dense. And within each canopy, these density types, I have measured the suspended sediment concentration and the flow velocity 5 centimeters from above the canopy bed. And also, I have identified a control location where in that place there's no any canopy present. And also, I measured the same parameters in that control location. So without further delay, let's discuss about the results. So when we analyze the suspended sediment concentration and the flow velocities within the canopy and in the control location, we observed very interesting results. So within the canopy, we observe fairly higher SSC values, or the suspended sediment concentration values, with respect to the control location. But in terms of flow velocities, we observe exactly the opposite. We observe fairly higher velocity in the control location than within the canopies. And also, when we analyze how the flow velocities and this SSCs varies among the different canopy density types, there we observe that the moderately dense canopy type seems to have the highest SSC values. And in terms of flow velocities, we observe that the moderately dense and the homogeneous dense, these canopies having a higher flow attenuations. And the sparse and the clusters density, the dense canopies having a lower flow attenuation. And also, when we analyze how the turbulence parameters varies among the different canopy density types, so you can see the clusters dense and the homogeneous dense. The higher canopy density type seems to have a higher turbulence production and dissipation parameters with respect to the low canopy density groups. And also, I would like to bring your attention to the SSC distribution happening among the canopy types. Here you can see the mean of the SSCs does not vary that much among the canopy types. But in terms of variability-wise, you can see the low canopy density seems to have a higher variability in SSC. And also, when we plotted the SSC within the canopy against in the control location, we observe that the low canopy density group seems to have a negative slope. So what does this mean? So this means that when the external sediment load increases at the control location within these low canopy density groups, the SSC started to decline. So this means that these canopies are shifting from sediment resuspension or erosion to sediment deposition when the external sediment load increases at the control location. However, when we look at the high canopy density groups, we observe the positive slopes. So this suggests that these canopies promote sediment resuspension when the external sediment load increases at the canopy at the control location. And also, we have calculated the sediment benthic remove rate for all the canopy density types. And from there, we observe that the higher canopy density group seems to have a higher benthic remove rate, or the higher sediment removal rate from the water column. So here you might be wondering, how come this high canopy density group having a higher sediment removal rate, even though they promote sediment resuspension? So in order to explain that, let me explain the sediment deposition and resuspension mechanism happening within these canopies. So as shown in this animation, so even though the low canopy density groups, even though they promote sediment deposition locally, due to the less turbulence happening within the water column, the already settled particles are not able to resuspended back into the water column. However, in the high canopy density groups, due to this special canopy arrangement, it will create wakes and generate turbulence, and these turbulence will bring already settled particles back into the water column from the resuspension. And also, unlike the low canopy density groups, these high canopy density groups have really complex structural arrangement. So within this canopy itself, there can be low energy pockets or the low energy zone that trap sediments. So that can be another reason for having higher sediment benthic removal rate in high canopy density groups. And also, with regard to the low canopy density groups, these high canopy density groups seems to have a higher live oyster population. So the biological trapping of sediment also fairly high in this high canopy density group. So that is another reason behind having a higher sediment benthic removal rate. And also when we analyze how the sediment mixing is happening within different canopy density types, here you can see a unique sediment mixing patterns happening among the different canopy density types. Specifically speaking, the low canopy density groups having a multidirectional mixing behavior, whereas the high canopy density groups having a unidirectional mixing behavior. This is mainly associated with the canopy, because in the low canopy density groups, the canopy height is fairly low. Because of that, the sediment have much more freedom to mix in the water column. But in the high canopy density groups, the canopy limit the mixing behavior happening in the water column. And finally, we have done a core spectral density analysis to quantify the contribution of turbulence and the flow velocities toward the sediment transport. So here, when you pay attention for the low canopy density groups, we observe that during the low frequencies, the flow velocity seems to have a higher magnitude toward the sediment transport. So during the low frequencies, the sediment transport is the main governing parameter toward the sediment flow velocity is the main governing parameter for the sediment transport happening within the low canopy density group. But when we are switching for the high frequencies, we can see the turbulence having a higher impact on sediment transport happening within these low canopy density groups. So here, these high frequencies are associated with the low water level, because the shallower the water depth, there will be a higher canopy water column interaction, and that will create wakes and generate turbulence. But once the water column started to increase, the canopy water column interaction will start to reduce, and because of that, the flow velocity will be the main dominant parameter toward the sediment transport happening within these low canopy density groups. However, when we are looking at the high canopy density groups, we can see that irrespective of low and high frequencies, the turbulence parameters seem to have a higher impact toward the sediment transport. So based on the results that we obtained from our field experimental studies, we were able to develop a framework to provide a better understanding about the sediment transport happening within these canopy types. So here you can see this framework is mainly consisted with two rectangles suggesting low canopy density groups and high canopy density groups. And we learned that the low canopy densities mainly trap sediments or promote deposition, whereas the high canopy density groups, they promote resuspension or the erosion. And within each rectangles, there are arrows pointing the dominant dominant mechanism toward the sediment transport under varying water condition. For example, if you look at the low canopy density groups, you can see during the high water level, which is suggested in the blue color arrow, it is pointed to the flow velocity. So the flow velocity will be the main dominant parameter toward the sediment transport. However, for the lower water levels, you can see the turbulence is the main dominant parameter toward the sediment transport happening in the low canopy density groups. And similarly, for the high canopy groups, you can see during both high and low water level, flow velocity, turbulence is the main dominant parameter toward the sediment transport. So having this kind of a structured approach is highly useful for oyster restoration activities, specifically for the coastal engineers or the coastal managers in terms of forecasting the sediments behavior happening under different canopy types or under different water level conditions. So that's all with my presentation. Thank you very much for listening. And yeah, let me know if you have any questions.
Thank you, Manisha. Thank you. All right, next up, as we try to get back on time, is Nathaniel, who's going to talk to us about 2 decades of citizen science.
Hello, everybody. Today I'm going to talk to you about a citizen science research project that has collected over 20 years of water quality data in the St. Lucie estuary and southern portion of the Indian River Lagoon. And I'm going to talk about how this data might be useful in calculating historical water quality trends and patterns. So in 1998, Florida Oceanographic Society has established a water quality program led by citizen scientists called FLOWS. It began with just a few volunteers that would go off to the end of their docks weekly to collect water quality samples. So over the years has evolved into this large research program with many, many dedicated volunteers that test at sites all across the St. Lucie estuary and southern Indian River Lagoon. And the main purpose of this project was to measure long-term water quality and capture temporal changes and spatial changes within our local estuaries, and also determine how our water quality was being impacted by environmental stressors, such as freshwater discharges, as well as the production of algal blooms. So over the last 20 years, our volunteers have gone out to their assigned sites weekly and collected an assortment of chemical and physical water quality parameters, which you can see on our table here. Some of the sites were public, while others are their private docs, which allowed us to expand our data range. And they tested at saltwater sites, brackish, within the brackish estuary, and then especially during the discharges, some of their sites were complete freshwater at some times. This led our data to be very variable depending on locations. So we established our FOS testing zones throughout the testing area that we had in the St. Lucie estuary in the southern Indian River Lagoon. We created 10 distinct testing zones based on different water quality characteristics, but it covers everything from the St. Lucie River to where it opens up into the larger estuary, and then finally where it pours into the inlet and the Indian River Lagoon. And so our volunteer sites have been dispersed all throughout these testing zones. Here's only some of our historical sites. Cumulatively, we've had over 110 active volunteer active sites. Some of them were only active for about a year or two, depending on the volunteers' commitment and participation, but some that were established early are still established today. And the application of this data mainly gets put into these weekly water quality reports that we send out. It is putting a letter grade to each one of our water quality zones. The letter grade is based on a criteria for selective parameters that just relates to general ecosystem health. And this is blasted via social media, as well as used in some of our advocacy talks. But we're sitting on this large archive of data that's been collected for over 20 years. Some that, it predates some of these other monitoring systems that we have active today. So we wanted to see, was there a way that we can use this citizen science data to examine historical water quality patterns and trends? And that was our first objective. But before we went gung-ho and building water quality models out of citizen science, first we would need to validate. It is citizen science. So we needed to validate 20 years of citizen science data. And how to best do that, one thought is we can compare it to some of the other monitoring systems that we have ongoing today. And this led me to develop my first question and the main question I'll be covering in this talk, but how does citizen science data compare to some of the other water quality monitoring stations? So we went out and we sought after different monitoring systems that are active today that overlap with our testing zones. First, we utilized the Erlon stations run by FAU Harbor Branch. It is a autonomous system that collects hourly water quality data every hour on the hour. We also utilized the Kilroy network run by Team Orca. Again, another autonomous system collecting on the hour every hour. And then finally, just to make sure all of our water quality zones had an overlapping reference system, we utilized the larger data set from South Florida Water Management District that collected field samples every month. So we had 3 reference systems that we were comparing against our citizen science data. So to properly compare it, first, we ran a Mantel test, a multivariate statistical test that allowed us to test the correlation between 2 distant matrices and data sets. We wanted to visually examine the difference in the sampling interval among the monitoring systems. And then finally, to examine how the data responds from each reference system to environmental stressors, such as freshwater discharges. So again, the Mantel test, it's a multivariate statistical analysis. It can be used to calculate correlations between 2 paired distance matrices. And the 2 main values that we got out of this test gave us a p-value, which tests for the significance, as well as a correlation coefficient, or R, to show us how related the 2 data sets were, either being a positive relationship above 0, a negative relationship below 0, or 0, meaning no relationship at all. So the results of this Mantel test are here. I'm going to walk you guys through it. So for each column is our water quality testing zone, and then each row is the reference system that overlapped with our water quality data. So for our first column, this is in zone 1. Anytime you see an empty cell, this just means our reference system did not overlap with our testing zone. But we can see that the district data was statistically significant, the comparison between the district and our volunteers, and that it was above 0, positively correlated. And as we flip through the rest of this Mantel test, comparing all of our zones against our reference systems that overlap, we can see across the board that every comparison is statistically significant, and they all have a positive correlation. Some of them more positive than others. You can see here the Kilroy compared to the FOS. At zone 7 and 8, it was below 0.5, so a little less positive than the surrounding test. But we're thinking this might be due because we had a data set from Kilroy that was only 2 years old, so not a lot of data points for comparison. But a little summary table just to show that over 70% of the comparisons were made were above a 0.7 R value. So to compare our sampling frequencies among our different reference systems, we used data just from this past year in 2024 in the same location, the Middle St. Lucie Estuary. So I'm going to show you a series of graphs. Each graph is going to be a different parameter that we looked at. Within each graph, you can see on their x-axis is time, y-axis is the parameter measurement, and then we have 3 different time series. Erlon in blue, which recorded hourly data, FOS in red, recorded weekly data, and data from the district, which is that single monthly measurement. And as we can see in temperature, that all 3 systems going throughout the year of 2024, they're pretty packed together and quite overlapping each other and follow this similar pattern. And we can see this even more so in dissolved oxygen, where all of the systems, for the most part, are very tightly packed together, following the same trends. Even with salinity down here, it being more variable, still following the same trends, maybe seeing a couple of monthly districts outside of the range. But this wasn't always the case. An example would be for pH. We noticed something really interesting with pH, that only during the winter months in January, February, and December that our weekly FOS data fit within the Erlon hourly ranges for that month. However, as soon as we got to the summer, there was a large disconnect, and FOS data was super elevated, which is really interesting. Finally, we wanted to test our system up against a stressor event, so we used the 2024 Lake Okeechobee discharges. This took place between mid-February and late March. And so to compare this, we used salinity as our parameter, and we looked at both our volunteers' data in red and our Erlon data in blue. So again, we have time on the x-axis, salinity on the y, and the 2 dashed marks indicate the first one is the start date, and then the second one, the end date of the lake releases. And as you can see, that both systems follow the same pattern. As soon as they hit that start date, both of them plummet to near 0. At this time, they were doing pulse discharges, so you can see that both the systems react to these pulses, and then as soon as we hit that end date, both the systems recovered at the same time with not much lag in between. So this is just the tip of the iceberg. We just first wanted to validate this. We found that our citizen science data was very comparable to the other systems, and from there we want to use this to model historical water quality patterns, looking at things like temperature and how much our estuary has altered from these stressors. And then finally, just wanted to leave off on a positive note. Citizen science is a great way for the research community to engage with the general public towards the same mission. And thank you.
Thank you, Nathaniel. All right, this is the last talk of the session, the second student presentation, and Olivia is going to speak to us about counting microcystis colonies using digital holographic imaging.
Hi, everyone. I am Olivia Rookty. I'm a PhD student here at FAU's Harbor Branch Oceanographic Institute, and today I will be talking about the rapid detection and enumeration of microcystis using digital holography. So harmful algal blooms are caused by the colonial cyanobacteria Microcystis aeruginosa, I messed up that sentence. Harmful algal blooms caused by the colonial cyanobacteria Microcystis aeruginosa are a significant and frequent global issue. It produces the toxin microcystin, which is threatening to aquatic life, human health, and the economy. Microcystis thrives in warm, nutrient-rich waters, making it a common problem in lakes, reservoirs, and rivers worldwide. And these blooms are notably prevalent in freshwater bodies such as Lake Okeechobee, which directly impacts surrounding ecosystems such as the Indian River Lagoon. Accurately measuring microcystis abundance is crucial because cell counts determine the true severity of a bloom. However, its colonial nature makes traditional methods ineffective for precise and rapid quantification. So our work addresses this challenge by using digital holography to rapidly and accurately measure microcystis colonies and their cell densities, with the intention of translating this work to in situ studies using holography. And this will provide a faster and clearer understanding of bloom severity, improving monitoring and management efforts moving forward. So in this study, we first start with an intact microcystis colony, as I showed in the previous slide. We image this using digital holography and confocal microscopy, which provide information about the shape and porosity of the colonies. And to accurately quantify microcystis abundance, we need to determine the number of cells within each colony. And since colonies vary in size and structure, direct cell counting is not possible without breaking them apart. So to do this, we sonicate the colony, applying high-frequency sound waves to separate it into individual cells. We then image these cells using holography, flow cytometry, and traditional microscopy to ensure accurate quantification of cells, and ultimately improves our ability to assess bloom severity. So going into the first of these methods, holography is a method used to capture detailed three-dimensional images. Here's a diagram of our lab holography setup. So our sample sits on that slide, creating diffraction patterns when illuminated by the laser beam, and that interference of the undisturbed beam with the scattered beam is what is recorded as our hologram. By reconstructing these holograms in 2D planes at different depths within the 3D volume, we can see each particle in focus clearly at each layer, and this process gives us a detailed view of where particles are located in 3D space within the sample. So for each experiment, we isolate a single intact microcystis colony. Then after imaging the colony, we break it apart with the sonicator and then holographically image the cells using a Palmer-Maloney slide, which gives us a fixed 100 microliter volume amount to relate the counts with the other methods. And here's an example of what an in-focus hologram looks like for a colony on the left and the cells that are on the right. And after sonication, each sample is run through the flow cytometer, which is used to measure the physical and chemical characteristics of cells or particles suspended in fluid. It involves passing these cells or particles in a single file line through a laser beam and detecting the light scattering and fluorescence emitted. As the light enters a cell, the laser beam is scattered forward and at different angles, which gives us our side scatter. And the amount of light scatter depends on the size of the cell and on the internal complexity. For this study, we're specifically using phycocyanin and chlorophyll A fluorescence to determine how much of our sample is actually a live microcystis versus what's just noise. In processing, we use k-means clustering to determine the ratio of live cells in our sample, which is then applied to holography-based counts for more accurate estimates, since holography can't distinguish between live and dead cells. And assessing this is crucial because dead cells do not contribute to the bloom's impact in the same way that live cells do. And by grouping similar cells into clusters, this allows us to effectively estimate this ratio, improving the reliability of our cell counts in holography and providing a more meaningful measure of bloom severity. The next technique used in our integrated approach is traditional microscopy. Using a compound microscope, a hemocytometer, and a tally counter clicker, we manually count the cells. We are also incorporating confocal microscopy into the experiments, which uses lasers to create 3D scans by scanning the sample point by point at different depths. Building an extensive set of scans will allow us to determine the average colony thickness for each strain, which will be incorporated into the data processing codes. So with the information we're collecting from each technique, it all works together to fully demonstrate the composition of microcystis. We have digital holography providing information about colony shape, porosity, and area using binary imaging that can distinguish the colony from the background. It's also used to determine the cell counts, but it cannot distinguish between living and dead cells, and that's where flow cytometry comes into play, which gives us the cell count and the ratio of alive cells, which we then integrate when counting cells in the holograms for accuracy. We also have confocal microscopy offering insights on colony shape, porosity, and thickness. And then lastly, traditional microscopy, which allows us to confirm the cell counts by doing them ourselves manually. So to date, we have processed 12 of 15 data sets and have completed 22 confocal scans. We're selecting a wide range of colony sizes, so the larger ones are going to have much higher cell counts. There is also great variability between samples expected, for instance, salinity, which measures how compact or dense a colony is relative to its outer boundary, with the values closer to 1 indicating a denser structure and the lower values reflecting a more porous form. And as you can see in this middle plot here, salinity exhibits a wide range of variation. Cell density and measurements from holography and flow cytometry show a strong correlation as most points align closely along the slope of 1. We'd expect a 10 to 30% variation due to experimental complexity. It seems that at higher densities, colony densities, there are more discrepancies with flow cytometry reporting higher values than holography, but overall the results demonstrate that holography can provide reliable cell count estimates. And it's too early to tell at this stage, but with enough replicates, we'll be able to determine the empirical relationship between colony size and cell counts. So our current and future work involves performing more replicates, and we also have been imaging microcystis from Lake Erie, which, as you can see here, is very morphologically different from Lake Okeechobee strains. We also have ongoing field work there, and we'll tie that into the study as well. The results from this study will enable rapid and accurate in situ characterization of microcystis cell counts during blooms using the AutoHolo, which is a custom-built in situ holographic imaging system that'll be trained to incorporate the empirical relations derived from this to characterize microcystis abundance and distribution from field observations. And importantly, we anticipate the empirical relation will be applicable to other imaging systems as well. And I'd like to thank the Indian River Lagoon Graduate Research Fellowship for the financial support. I'd also like to thank Dr. Aditya Nayak, Dr. Malcolm McFarland, and Dr. Karuna Agarwal for their great support and guidance throughout this research. I'd also like to thank the whole Nayak lab, but especially Dr. Karuna Agarwal and Madison Bennett for their immense help in processing data and conducting experiments. And thank you all for your attention. I'd be happy to take any questions if we have time.
Yeah, we'll take one quick question while Irene, aka the enforcer, makes her way up here to take charge of the next session. Is there a question? You might be off the hook.
Okay, thank you.
All right, we're going to start with the
We're going to start with a 3-minute, 3 slides. First up, Bob Vernstein, best seagrass planting success.
So why do we plant seagrass? And I ask that not as a smart-ass question, but as a scientist, I think sometimes it's more important to ask questions than to answer questions. And you'll notice that my title and the subtitles are all questions here. But on my soapbox first, if conditions were right, planting seagrass may help accelerate recovery of seagrasses. But it's not sufficient. And if we really want to recover seagrass, we need to reduce nutrient loading to the lagoon. So I think planting seagrass is still in the research phase, and I'd like to address some of those questions. There are a lot of questions about planting unit size, type, anchorage, spacing, plot size, and I want to address one of those questions. So, for example, what's the best strategy? If You might think like, if you thought like a plant, you might think, well, if I'm in the center of this big patch, and I couldn't draw a whole big bed, but if I'm in the center of that, a grazer comes along and I'm sort of protected, but at the sacrifice of lots of my neighbors. An alternate is, if you think like a grazer, that patch might be the big salad bar. And I don't have a pointer? There it is. Only if you're a little patch way away, you may survive. So let's look at a couple examples to address this question. So I'll show 3 scenarios of plantings. This first one is a 1/4 acre or 1/10 of a hectare at half-meter spacing. That's 4,225 planting units. And it might look like that. It's a 50-meter dock just for scale. An alternative with the second one is maybe planting 2-meter by 2-meter plots. And again, on half-meter centers, that's 25 planting units per plot. And to scale, it might look like this with the same number of planting units. I get 169 plots that way with the same number of planting units. A third way might be to plant a plant in the center and 4 plants a half-meter away from that. And this is something that I think volunteer citizens could do. You mark the center with a stake and plant in an X or a T pattern. If you do that, you could have 845 plots. And then I'll need to ask, which is best? And I don't know. And I don't think anybody knows, really. But considering that failure is a good teacher, which of these strategies might we learn the most from?
All right, we're out of time.
Okay. Well, in memoriam to Dennis, this is Dennis and I probably discussing whether seagrasses have strategies.
Thank you.
Okay.
Up next, we have Christopher Moore, Parasite Diversity of Crustaceans and Fishes.
Well, my name is Christopher Moore. I'm currently a postdoc at the University of Florida. But in the next 3 minutes, I hope to share with you some results that we've currently got in review now at Estuaries and Coasts from my time here with Mike and Krista McCoy as a postdoc. So parasites. So we're particularly interested in trophically transmitted or multi-host parasites. So these are parasites that require multiple invertebrate and vertebrate hosts to complete the life cycle. You may start off at an obligate mollusk first intermediate stage. Those parasites at this stage can be swimming and swim out and infect a variety of second intermediate hosts where they would become dormant and form cysts, mud crabs or fishes. And then this broader arrow here denotes trophic transmission. So for the parasite to complete its life cycle, it must be consumed by an appropriate host. And so I want to leave you here from this image with the idea that without any of these hosts, we won't have these parasites. And rather than represent disease, these parasites actually, in my view, represent trophic interactions and trophic complexity. So we wanted to kind of bring this idea to the IRL and use it as a monitoring tool. In the face of catastrophic losses of seagrass, as restoration continues apace, can we use parasite diversity to monitor the restoration of trophic complexity in seagrass-associated food webs? To do that, we needed to collect some baseline parasite data, which we did using these handy, what we call crab condos, these sampling devices that we put out in seagrass areas throughout the IRL. Sampling for those benthic fauna at the base of food webs that accumulate a lot of those parasites, those mud crabs and fishes. And so we kind of tapped into the second intermediate host stage here, dissecting about 800 xanthidae, mud crabs, stone crabs, about 400 porcelain crabs. And we also dissected a number of cryptobenthic fishes that serve as both second intermediate and final hosts for parasites. Because this was baseline parasite data, we didn't have anything to compare this to, so we used meta-analysis to provide context for our work here, comparing our data to data from the 29 other studies throughout the globe, comparable estuaries and coastal systems using comparable taxa. It wasn't really a surprise to us that parasite prevalence here in the IRL, shown in salmon color, was lower than that reported in the literature, shown in teal across a variety of metrics. It was a little surprising to me to see how low some of these larval stages were of these parasites that had implications for trophic complexity. We were missing, in some cases, complete parasite groups from crabs and fishes. The IRL, according to land cover data in our area, is a suburban environment according to population density and the built environment, so much lower here compared to other suburban environments sampled in the literature, but in fact was more comparable to urban environments here. So it's lower. Our IRL parasite prevalence data is lower even than urban environments, but closer to urban environments than suburban environments. And so this just echoes, I think, Bob's comment here that when we're focused on restoration, we need to be focused on what's happening in the watershed. That in turn determines.
Okay, we're out of time.
In the lagoon. Boom! Nailed it.
Okay, up next we have Carl von Schneider, Evaluation Protective Materials for the Growth of Mercenaria in IRL.
Hi there.
Please stay at the podium because people online can't hear you.
Okay. Hi, I'm Carl von Schneider, Conservation Restoration Coordinator with Brevard Zoo. Brevard Zoo's Restore Our Shores program has implemented clam restoration projects in the Indian River Lagoon since 2021. Since then, we've had varied success due to water quality, location, and methods used, but our main issue that we wanted to address was predation. So we, spring of 2024, we partnered with Brevard County Natural Resources and Richard Baptiste at Harbor Branch and received 1 million seed clams, which we distributed across 20 sites in the central Indian River Lagoon in southern Brevard County. And 5 of those sites received additional protective materials to test. So all 20 sites received polyester mesh bags. These traditional clam bags, they limit predation, but they allow crowding, and they also foul very easily and must be changed to a larger diameter mesh size as the clams grow. So the 5 out of 20 sites received bottom planted clams with quarter-inch plastic mesh on top. This limits crowding but increases predation of the seed clams at their most vulnerable state because smaller predators like crown conchs and blue crabs can still get under and even eat the clams through the quarter-inch holes. And as a natural non-plastic alternative, we covered bottom-planted clams with coconut fiber. And so what did we see? We saw that polyester mesh bags had the most seed clams surviving after 5 months at the 5 sites, and followed by bottom-planted clams with plastic mesh. And there were little to no surviving clams under the coconut fiber due to easy degradation and wave action, and it's probably not a viable option. So, what's the takeaway from this? So, obviously, the polyester mesh bags are the best way to grow out seed clams in the lagoon and for aquaculture purposes, and followed by bottom planting with quarter-inch plastic mesh, and coconut fiber was missing. So what's the take-home? The take-home is that I would like to grow out the seed clams in polyester mesh bags until they are large enough to bottom plant and use the plastic mesh as a cover so that they can grow more so that they grow to maturation and eventually spawn, which is the goal of clam restoration. Thank you.
Thank you for that. Next up, we have student presentation. We have Emily Sermon. Are we better with age, natural versus artificial reefs?
some things we know get better with age. For example, cheese. We know exactly how long it takes to age cheese to produce specific outcomes. Unfortunately, in the field of restoration ecology, things are far from that simple, and we have longer timescales for ecological recovery typically than what we are able to monitor. So my research aims to understand how aging artificial reefs compare to natural reefs in health, persistence, and habitat provisioning in the Indian River Lagoon, the southern Indian River Lagoon, and St. Lucie Estuary. This past summer, we started sampling multiple oyster reef health metrics across 3 age categories: 6 to 8-year-old artificial reefs, 9 to 13-year-old artificial reefs, and natural reefs. And we did metrics across multiple different health metrics. However, I'm only going to focus on 2 today: reef composition and density of live oyster. So for reef composition, we placed 6 random quadrats on the reef, 1 meter squared grid quadrats, and we counted and tallied each substrate that occurred under each cross string. And then for density of live oyster, we counted every live oyster over 10 millimeters in size within 3 of those 6 quadrats. So our preliminary analysis shows that despite their mature age of all those age categories, age category still does seem to influence reef composition and density of live oyster. So for reef composition, you can just think of blue or cool colors as good and red or warm colors as bad. So dark blue is our eastern oyster, our target species, that we're hoping most of these reefs should be primarily composed of. Meanwhile, that sort of salmon color is shell culch. So that's what the artificial reefs are originally made out of, and also not so great as an indicator of reef health. And you can see there is a statistical significance between the 6 to 8-year-old artificial reefs and our other 2 age categories according to our Permanova and pairwise comparisons. Then when we look at density of live oyster, we see the same pattern. So again, we have those different reef categories on the x-axis, and y-axis is our density of live eastern oyster. And you can see again, there's not a significant difference between the 9 to 13-year-old age group and the natural reefs, but there is a significant difference between that, quote, younger age category, even though they are all mature, and we sort of expected them all to look similarly. So in summary, artificial reef age does seem to influence reef health and persistence even when all these projects are quite old, but further research is required. I would say this is the definition of preliminary data, and everything could be flipped on its head as I continue analysis, looking at salinity regimes, reef area, and we are going to be doing more sampling next summer, looking into habitat provisioning as well. So like I said, very preliminary. This is all part of a larger project resulting in a quick assessment monitoring guide for the southern Indian River Lagoon and St. Lucie Estuary as a collaboration between University of Florida, Florida Oceanographic Society, and the aquatic preserves. Thank you.
Perfect timing. Up next, we have Lorraine Simpson, Cajun Seagrass Restoration Projects.
Hi, good afternoon, everyone. I am Lorraine Simpson. And I'm sure I don't have to explain to most of you that there's a lot of seagrass restoration projects taking place in the Indian River Lagoon right now. And in some of those projects, practitioners are utilizing herbivory exclusion devices to hopefully increase project success. However, there's not a lot of directive or guidance on whether we should be doing this or how we should be doing this. And so my collaborators and I decided to jump into the literature and try to collate the current state of the science. So we did this by employing a meta-analysis following the PRISMA guidelines. And a meta-analysis is essentially a quantitative review in which you test the effectiveness of a variable, a treatment variable, such as caging here, by calculating the weighted pool estimate of the outcome. And our outcome is any change in seagrass growth or productivity in a specific population. And our populations that we targeted were our canopy-forming species that we find here in the Indian River Lagoon. So essentially, we were trying to collate any sort of literature from researchers in which they did an experimental or an observational field study that they collected seagrass data in a cage versus control treatments. And so we pulled together a search string with all those words I just mentioned and put them into several different databases, and we came up with 494 hits on papers. There were a few duplicates in there, so we screened 487 of those for keyword, abstract, and title. Once we started screening, we realized, well, we kind of missed the mark a little bit. There's a lot of papers in here that don't match our criteria, and we only had to actually pull 33 papers for a full text review. Once we went through and read all of those full texts, we realized that, wow, a lot of people don't put the information in there that we need, mainly means, and standard deviations or standard errors. Please report all of that data. So because of that, we had to exclude 21 papers, leaving us with 12 for this meta-analysis. So for this meta-analysis, of those 12 papers, only one of them actually was a restoration project. All the others were an experimental design looking at trophic cascades or nutrient cycling. There was a myriad of different K-gene methodologies. There were 21 different sites across those 12 papers, and they reported outcomes for our 3 forming canopy species, with Thalassia being the one that was most reported on. And out of our 6 different outcomes that we identified, there was 165 different responses. And so from the responses, we pulled those means, we pulled the standard errors or standard deviations, and we calculated our effect sizes. When we calculated our effect sizes and did our meta-analysis, lo and behold, nothing was significant except that of leaf area. But if you look at the numbers within the parentheses, those are the sample sizes, which are very low. And because of that, this calls into question the power of the test, as well as increases publication bias. And so it really further highlights the need for more robust studies to strengthen these findings. And so because our quantitative review was not what we wanted, we are asking everyone to help us do a qualitative review. So if you're a practitioner, please scan this QR code and do a survey.
Okay, next we have Lori Morris.
Is that all you have to say?
Seagrass, macroalgae, and phytoplankton. Get the net ready.
My hat that Dennis gave me, so I remember not to say bad things. So, okay, so I have our classic seagrass map, our data up there. Just mostly I want you just to see, just to remind you all that you haven't seen it enough, the trends of seagrass. Up, down, and up again, right? But believe it or not, seagrass is not the only primary producer out there. And so my question is there a balance, and how are the 3 seagrasses, drift macroalgae, and phytoplankton kind of competing with each other to keep the balance of the system? So in the picture, you can see seagrasses and drift macroalgae coexist well as long as we have good water clarity and conditions. But let's take your vision up here to the classic graphic that goes back pretty far. And if you have a good system with good water quality, good nutrients, low nutrients, and good light, you'll have a seagrass-dominated system. But as that changes, as the nutrients start increasing and you start getting decreasing light, the system can flip from a seagrass-based system into a phytoplankton system. And so this is exactly what we saw happen out in the lagoon. So if I take our seagrass and our drift data from our transects, convert that to carbon, take the carbon data that EdFlips collects from UF, from the phytoplankton, and put them together, you can see I can create the same graphic. And over time, in the beginning, in the 90s and the early 2000s, we had that seagrass-dominated system. Then once those blooms started and the blooms continued, we had a phytoplankton-dominated system. So looking at that graphic, if you kind of take it, and if I hypothetically stacked all those 3 primary producers on top of each other, it almost looks like there's like an amount that the system can handle, whether who's in charge, who's got the predominance. It's either seagrass or phytoplankton. But it doesn't make sense. Even though we're seeing seagrasses increasing here, phytoplankton's been low the last couple of years. So I'm not able to fill that line. I can't reach that purple line. So what is kind of, we're trying to figure out what's out there that can balance out that system. And what we're finding is probably out there is Caulerpa prolifera. We know there's dense Caulerpa beds in the southern part of banana, central banana, southern banana, and central IRL. And that is taking up that niche in that center area. And so I'm going to kind of leave you hanging. So I'm not going to finish the story because she's going to start beeping. And you got to come back in 2 weeks to the Sears meeting. And so I'll finish telling you the story then. And that's our great plan.
3 seconds to spare. Okay, next up we have a student presentation. We have Mara Scadden, Benthic Community Response to Shoreline Restoration.
Hello, everybody. You have 9 minutes left. Bear with me. My name is Mara Skadden. I am a doctoral student at the Florida Institute of Technology with Dr. Emily Ralston, and today I'll be presenting a very small piece of my dissertation in 3 minutes. All right. So this project took place at Carrs Park, which is located in the southern end of the Kennedy Space Center complex in the Banana River Lagoon. As you can see from this picture, we had some issues. So there was wave action, which caused erosion, which compromised the roadbed of this shoreline adjacent road, which is important. There are Kennedy Space Center buildings nearby. They got to use it. And so they had to move forward with some construction. And this team decided to move forward with a hybrid living shoreline, meaning that it had some sort of man-made aspect. And in this case, we were looking at coquina riprap and coquina wave breaks. And so the coquina riprap was only placed in about 300 feet of this section. The rest of the shoreline was left natural, but these areas were more significantly impacted and needed further protection. But you can see shortly after the deployment of this site, we had a natural sandbar start to occur behind those wave breaks. And so data collection for this was taken outside the wave breaks at approximately 15 feet from mean high water with a Ponar grab. They were taken in triplicate, and there are 6 zones of experimental sites and a control site. And shortly after deployment, we saw natural recruitment of all 3 species of mangroves, which is incredibly positive. The Kennedy Space Center staff also dutifully raised and then planted mangroves during construction and then planted them after behind the riprap, and they did wonderfully. More pictures of what it looks like, but basically, when we're looking at that pre-data, this site was not unhealthy to start with. It was okay. We had generally a decent species richness and diversity, or in abundance, but the diversity was low. We had generalist polychaetes, or the annelid worms, and some amphipods and crustaceans. But after restoration, we saw a massive bloom in Rupia seagrass. Woo! So this was a bare, sandy bottom before. There hadn't been seagrass prior to construction. And so we have beautiful, dense Rupia seagrass that was doing so well, it actually bundled up on top of itself and got sunburnt. Which it's okay. But these are our new diverse community that has come in associated with that restoration and that seagrass coming back. And when you look at our Shannon Weiner diversity indices, you can see that especially in the spring of 2024, we had a massive increase in diversity at these sites. When you see in the control that we also had a minor increase there, but it's statistically significant in the experimental sites. Yay, taxonomic diversity. You can see that the species richness stayed about the same, but that's when we're switching from, generalists to the specialists.
Okay, time is up. Thank you. Okay, up next we have another student presentation, Mariam Sanbal, Habitat Association and Abundance of Bivalves in Mosquito Lagoon.
Hi, everyone. Bivalves are a critical component of estuarine food webs and are included in the diets of some of our favorite animals, including the black drum, blue crab, cow nose rays, and of course oyster catchers. It's well known that these predators consume the hard clam and eastern oysters, but other smaller and thin-shelled bivalves are consumed as well. Because these bivalves are not commercially valuable, they're understudied, and not much is known about the populations and the habitats associations in Mosquito Lagoon. The last biodiversity survey of bivalves in Mosquito Lagoon was published in 1995, but data was minimal. So we sought out to get a preliminary look into the community dynamics and habitats of bivalves in Mosquito Lagoon. To do so, we sampled 12 sites, 4 shoalgrass beds, 4 unvegetated sites, and 4 sites used for shoalgrass restoration. At each site, 4 quadrats were haphazardly sampled for percent seagrass and epifaunal bivalves, and within each quadrat, 1 sediment core was taken to sample infaunal bivalves. Additional abiotic data was also recorded, however, I don't have time to go over those. Sampling was completed twice, once in March and once in May of 2024, and unfortunately, hurricanes Milton and Helene precluded sampling in the fall season. So we found a total of 44 juvenile-sized bivalves across all sites and all sampling periods, and our results found using generalized linear mixed models to account for zero inflated data. We found that the nuclear proxima had the highest average abundance across all sites and sampling periods, and the Chrysostria virginica abundance was significantly higher at shelly restored sites because the shelly sediment was acting as substrate. And then seagrass habitats did not support higher diversity, contrary to our expectations, and compared to non-vegetated sites. And our key takeaways from this are our sites have consistently low abundance and diversity, and while our data is minimal, it's a preliminary look into the present bivalve community in Mosquito Lagoon, and further monitoring can provide insight into community shifts as environmental conditions change. Thank you.
"15 seconds to spare." Okay, next up we have Beyond the Unknown, revealing microturbellarian biodiversity in the Indian River Lagoon.
Hello everyone, my name is Yander Diaz and I'm postdoctoral research on the Smithsonian Marine Station at Fort Pierce. Microturbellarians are an heterogeneous group of flatworms that basically are defined on basis of their size because they are the smallest one of the group of the flatworms, and also because most of them, or almost all of them, are free-living species or endo or ectosymbiont. Therefore, they are not a taxonomical group, they are an ecological group. You can find them in all kinds of ecosystems, always associated, of course, to the water. They cannot survive in completely dry systems. However, some species are limnoterrestrial ones, and they can live in wet soils and also associated to mosses. And they are very important because they prey and control a lot of other microturbellarians or other groups of invertebrates in marine and freshwater ecosystem. If you see to the flatworm in the top, you can see a tubular structure in the middle part in the posterior side of the worm. That is the male copulatory structure of a flatworm, but it's not this species. It's another species of flatworm that was prey for this species. And in the flatworm of the bottom, you can see that the gut lumen is completely full of diatoms, microalgae. The picture I found before to come to here to develop my project is that merely 165 species of marine turbellarians are recorded to the coast of the United States, both the Atlantic and the Pacific coasts. It's not that low considering that they are 10% of all the species described worldwide. However, it's very dramatic when we move and take a look to Florida, where only 6 species of marine microturbellarians are recorded, and from these 6 species, only is recorded to the Indian River Lagoon. Therefore, the focus of my project is documenting, cataloging the diversity of microturbellarians of the Indian River Lagoon in terms of their taxonomy and their molecular phylogeny, and also describing the spatial and temporal distribution of these species. In 1 month of work, I moved recently to here, between January and February, I have found here 110 species. 74 of them are rhabdoceles. Rhabdoceles is a group of more skillet, and therefore I know that more or less 50 species of them, 70%, are pretty new to science and need to be described, and the other belong to other groups of microturbellarians. Of these 110 species, only 1 was recorded before to the Indian River Lagoon, that is Phonorhynchopsis hegeni, the picture in the top, and the other one also recorded to Florida. The 2 records for Florida is Orostylis timocorum that was described from northern of Florida. Thanks for your presentation. Very few results, but I'm here for the last month only. Thank you.
Wonderful. Thank you. Can we have a round of applause for all our presenters for the day? So before we start with our closing remarks, I'd like to ask the poster presenters to please get out of here and go get some food before everybody else does. And then we're going to start the poster session. If you are one of our judges, please make your way over to the posters that have been assigned to you. We'd like to have all the judging forms submitted by 6. so we can tally up the scores. 7:30, we will have our award ceremony. Please remember to vote for your People's Choice Student Awards. These mean a lot to our students. If you are a judge, please be sure to write comments on your judging forms because those will help students in future presentations.
If you are politely waiting for closing comments, Irene actually covered everything and there are none right now. But please come back at 7:30 for the student judging. Sorry about that. Another note, the food is not yet ready, but the bar is open, so you know where to go.